And you also have therapy for anyone who's had like allergy shots or allergy tablets. This is what that is. And so you may be thinking, there seems to be a lot of biologics, a lot of blue on the board, so maybe we're good with asthma. But actually, there's still a lot of unmet needs for this disease. So if you look here, this was from a recent review in Lancet. And it's showing you that while there are a lot of biologics out there, for some of them, or for many of them, you do need to meet some clinical criteria to be eligible to receive these medications. Now, one exception here is anti-TSLP, again, something that acts way earlier in the cascade. Aside from the need to meet certain clinical criteria, these drugs are also pretty expensive. So it's cost prohibitive for a lot of individuals, and a lot of them are also just add-on therapies. And then we do have the therapies that will completely change the course of the disease. So for example, allergen immunotherapy acts here at the level of DC, and it can basically deviate the response away from type 2 immunity and basically be a curative treatment. Now, the problem is it does take a long time for people who are undergoing this. I don't know if you know anyone who's on it. They have to do these shots, initially, at different frequencies, but it's basically a regimen of 3 to 5 years. And then there's the potential for adverse events like anaphylaxis. And then some of the more recent ones that are coming, for example, the allergy drops, that one's not really covered by insurance. So there are still a lot of roadblocks, and there's still a lot of need for new targets and new therapies for asthma. So how did I get into studying allergic asthma? I wasn't always studying this, so in my postdoc, I was actually studying this pathway and this receptor called NOD2. And NOD2 is actually a receptor for bacterial peptidoglycan. And this receptor binds, once it binds peptidoglycan, it will bind and activate this kinase called RIP2. And we found that this kinase, we and others have found that this kinase is really crucial to transducing a lot of the signals emanating from NOD2 and activating all of these downstream pathways. We also found that because it's kinase, it's very druggable. And so we got interested in this project because there was also some reports that had come out showing that this receptor promotes type 2 immunity, basically the allergic type of response. And so we said, well, why not RIP2? Let's look at RIP2 in that scenario, and if it is involved, then it might be a potential target for allergic asthma. So a lot of this work was done by a previous graduate student, Maddie, and supported through a Canadian ROO. And in our model, I just wanted to briefly show you what we do. We basically have dust mite extracts. We intratracheally deliver that into mice. So we have some wild-type mice and some mice which lack the RIP2 kinase. And then we deliver that in day 0, day 7 to 11. We sacrifice them at day 14. And we can measure the lung function, do a lavage to look at the cell subtypes and flow cytometry, and then also take the rest of the lung for histology. So obviously, in the interest of time, I don't have a lot of time to show you all the data, but this is, I guess, just the most striking histology from there. And this is a wild-type mice and RIP2 knockout mice. Then we have on the left of each naive mice or mice which we've put under the asthma model, the dust mite model. And you can see that there's a significant reduction in the airway inflammation when RIP2 is absent. So suggesting that RIP2 does promote dust mite-induced lung pathology, or does promote dust mite-induced allergic airway inflammation. Now, Maddie followed this up because we knew it was a kinase. We had found inhibitors for it. We partnered up as well with GSK, who had some spare compound that they weren't going to put forward. So we got some compound from them, put that into rodent chow, and we're able to basically pharmacologically inhibit RIP2 during the very beginning sensitization step. So you can see it's just around this first period, and then the rest of it, RIP2 is not inhibited. And even just with that one treatment, you can see that there's a dramatic improvement in the airway inflammation in these mice. And so just to summarize a lot of years of work, either genetic loss or pharmacological inhibition of this kinase, and we think that it's at the level of the airway epithelium that leads to a reduction in a lot of these epithelial-derived chemokines, which reduces the airway inflammation and pathology, as well as the type 2 and type 17 responses. We did find, however, that this had no effect on airway hyperresponsiveness. So affects inflammation, but not necessarily the hyperresponsiveness of the lung. And so I'll take a moment to just kind of switch gears. So now I'm switching into a new area. I mentioned previously that these drugs or these therapies which act very early on, so tezepelumab at the level of TSLP, allergen immunotherapy likely at the level of the dendritic cell, these are actually the more efficacious treatments. So versus all of these others that act downstream. And so we were really interested in these very early events that occur in recognition of the allergen, because we felt that might have the more profound effects. And so this work was initiated by Maddie, continued by another student that had just defended, Lizzie, supported by American Lung Association as well as the NIH. And to summarize a lot of work, basically we conducted a screen to find novel receptors for house dust mite. So house dust mite is one of the most common allergens associated with allergic asthma, so we thought that's a good place to start. And what we discovered from that screen was this receptor called LMAN1, or lectin mannose binding 1. And so the interesting thing was not that this protein was not described before, so it actually also goes by ERG53. It was already known as a receptor. However, it had never been really described in the context that we are describing. So it is not known to be to have a function in signaling or have a function in immunity. And it was really more known to be a cargo receptor, shuttling proteins between the ER and the Golgi, maybe trafficking proteins out of the cell. And it was called lectin mannose binding 1 because it has this domain that recognizes monosylated proteins, which we think are present in allergens. And through a lot of data, again, not have the time to show you all, but basically we found that at least in vitro, when we looked at cells and epithelial cells, this receptor seemed to have a negative regulatory role. So we think it might have been dampening the immune response to dust mite, and this is just flow cytometry showing you the expression of LMAN1 on the surface of dendritic cells, peripheral dendritic cells on healthy controls and asthmatics, showing that it's significantly lower in the asthmatics. So I didn't have time to show you, but this is our model. And what we think is happening is that early on, you have other components of the dust mite that are acting through activating receptors to initiate signaling cascades resulting in activation of NF-kappa B, driving production of a lot of these cytokines. But later on in the response, this receptor LMAN1 comes in, and it recruits a phosphatase, which then shuts off a lot of that signaling. And so at this moment, we don't know, is it a cause or a consequence that you have lower expression of LMAN1 on the dendritic cell, but you can imagine a scenario that if you lose a negative regulator of inflammation, you might end up with more inflammation in the setting of asthmatics. All right, so what I hope to have shown you today is that there's a potential for targeting these molecules, for example, with the use of RIP2 inhibitors or RIP2 targeted. Now there are PROTACs, so basically degraders of RIP2, acting at the level of the airway epithelium that seems to be beneficial. And this one we haven't tested yet, but potentially You could think of using LMAN1 activating antibodies or overexpressing this receptor at the level of the DC and maybe potentially also deviating this response away from type 2 immunity. And so I would just like to end there, show you some pictures of my previous students, and here's the current lab. Thank our funding sources and all of you for your attention. Thank you.
Wow. Perfect.
Did it on time.
We were early. Yeah, so we have time for questions.
I'll put it back to the hall.
Anybody?
Yeah.
Siggy.
Any clinical tests, human volunteers?
No, so that's interesting. So for the RIP inhibitors, they're actually more interested in looking at that in the setting of inflammatory bowel disease because the receptor upstream of it, there are polymorphisms in those which are associated with Crohn's disease. So they're really more interested in looking at that in IBD, and they have gotten into clinical trials and they had to halt it. They didn't say exactly why, but something to do with toxicity. Yeah.
Wow.
Yes.
Frank?
So I have kind of a related question. What's the situation in IBD versus this with some of these inhibitors or with LAM1?
For LMAN1, yeah. So for IBD, so that's a good question. So in IBD, basically all or numerous animal models of inflammatory bowel disease have shown efficacy, when you inhibit RIP2. So they're really excited about that area. And this is kind of where, the PROTACs also started coming in. So in terms of LMAN1, nobody really knows. So in humans, there are mutations in it. Really, they've looked more at, it also shuttles factor 5 and 8. So these people have factor 5 and 8 deficiency. Like, according to someone who works with them, he said it's kind of mild, it's not anything life-threatening. So, in that case, if this is our indication, though, we're actually not looking to inhibit it, but maybe promote expression in particular cell types.
Questions? Oh, I have a question.
Yeah.
So I see here that most of the drugs are like monoclonal antibodies.
Mm-hmm.
So is there a chance to like make a cocktail of these and maybe test
there are
it?
some
Yeah.
drugs that are, I think that work on both, on 2 of them, at least at a time. I forget which one that is.
Yeah.
But still, what they've seen with all of these downstream therapies is that it's really important to stratify your patient population. And if you can really show, that's why the indication is such, like for their eligibility to be on these medications, is because they showed like, okay, if we really stratify, initially when they did the trials, they said, oh, there's no effect really, but then they tried to stratify the individuals receiving it into, what is their blood eosinophilia level, what is their IgE level, and only when they did that, they saw, okay, only these patients who are really high actually benefited. So, so basically, once you get the cascade going, it's a little bit harder, I feel like, to actually.
Yeah, because I was thinking, since you have a lot of potential targets, what if you mix, the drugs, so you are hitting more than one target? Yes.
So is asthma an early disease? How early does it start? And can you sort of like prevent it or?
Yeah, so for type 2 asthma, there's different, again, within the type 2 umbrella, there's many different types, but there's a majority of allergic asthma will be kind of like an early onset. I'm sorry, what's your second question? How?
I was just wondering, could you then prevent it? Is it a development?
Yeah, well, the thing is that when it starts really early, like for myself, for example, like you can't outgrow it, right? So sometimes maybe you'll see it in children, and then as they change, and it is known that sometimes hormones can play a role, and this will change basically severity of the disease, exacerbations, all of these things. but some people outgrow it, some people develop it later on, and sometimes the ones that develop it later on have a more severe form, not so amenable to the conventional therapies.
Not a question per se, but I just want to acknowledge what you're doing. Like, you're discussing asthma. I am very familiar with IBD. And actually, when I saw the last three letters MAB there, the two things that I thought right away were infliximab and ustekinumab, both produced by Dr. Villalobos, J&J. And they're very, very expensive if you don't have
insurance. Right. Yes, for sure.
So not a question, but
yeah.
I just want to get your...
So definitely, so when people like say like, haven't we solved that already? We have so many drugs for it, isn't that fine? If you could target it with some small molecule inhibitor, for example, that's a lot cheaper than monoclonals for your targets. Yeah. Yes.
So RIP2 is that RIP2 kinase is one. So is that the same pathway?
Yes.
So just to know that kinases would be a druggable target, easier to design drugs that would need with kinases. So how would that affect our pathway? We were to the RIP2 kinase.
Oh, that's what we were drugging. Yeah.
So because it says RIP2.
So RIP2 or RIP2K, the same thing. Yeah.
It's the same.
Yeah.
One is RIP2, the other is kinase.
Oh, I think you're talking about like NOD2.
Yeah.
So NOD2 is the receptor that's upstream of RIP2. Some people have also tried to drug the receptor itself, and with some success, but really not as good as if you inhibit the downstream kinase RIP2. Yes.
Not a question also, but a comment, because it seems like there's a lot of things going on with the high mannose glycans because of the LMAN1. I mean, we were also looking at cancer before, and we found a high mannose, like elevation of high mannose glycans in the surface of the cells. So I really think there's something going on with the N biosynthetic pathway, that there's a deletion going on, that it cannot proceed to make the complex and hybrid glycans anymore. Just staying there because that's in the beginning of the N biosynthetic pathway and glycan biosynthetic pathway. So I thought that was really cool that it kind of
I actually don't know how different the pathway is on, because obviously what it's binding is going to be the dust mite proteins, right? So I don't know how different the way they make their glycan modifications versus we do, but at least there are a couple other studies that have been looking on glycosylation of allergens, and it seems a fair number of them are glycosylated and specifically monosylated. So what we actually are thinking is that, yes, we are currently we are funded to kind of look at this receptor in the context of other allergens as well, to see if it can be, more universal, which would, not limit you to just dust mite allergies, but maybe any allergen which has a lot of this mannose.
Are we done?
Thank you so much.
Thank you, Justine.
Do you want to just look up here for me? Oh, is it here?
Is it on this one?
Yeah, Mario's uploading it. Yeah.
And then I'll use this computer over there. Is that what?
Or back up.
How will I move the slides?
Do you have it there?
No, it's easier to have it.
Yeah, maybe I'll just.
Yeah, just bring
Sorry,
your.
Steve, we'll take that back.
Okay.
Yeah, because this is a different. Yeah.
Plug it in later. We'll get it in first.
Yeah. Okay, our next speaker is the husband. Can you tell this is like a family affair? Yesterday was the mother and or mother-in-law.
Yeah.
Okay, we have George Aranjuez, PhD, also from the same institute, University of Central Florida, Burnett School of Biomedical Sciences, but he's into immunity and pathogenesis in the College of Medicine. Can I have the bio? Can I have the bio notes, please? Bio notes.
Notes.
Oh, yeah. Ako na pala.
Down. Yeah. Scroll down.
Scroll down.
Yeah, there.
Yeah, but it's not.
Scroll down.
Sorry. Not responding.
Yeah, I was trying
So what?
to show that she needs to
You were
to
showing
read
your
this.
screen?
The bio notes.
Yeah, you were showing your screen over there.
So I
Only I can control it. I can't. For some reason, I can't. It's not the right one. It's not in sequence.
Oh, okay, then I don't. Do you know which one?
Huh,
Do you know where it is on the slides? Oh.
Where? It's not in order.
Yeah, we don't know.
That's not him, wait. No, this is Frank. Okay, thank you. Okay, to introduce George, George Aranjuez graduated magna cum laude from UP Diliman with a BS in MBB, Molecular Bio and Biotech. He received his master's degree in neurosciences and then his PhD degree in genetics and genome sciences from Case Western Reserve University in Ohio. And then he did his postdoc training in cell biology in the context of infectious diseases at the College of Medicine, University of Central Florida. So he's shifted fields, a bit. So genetics, cell biology, and then host-pathogen interactions while studying multiple bacterial pathogens of public health concern. So George Aranjuez is finishing up his postdoc training. He's looking for a job. Hire him.
So listen first.
Yeah. So without further ado,
Thank you for the introduction and the opportunity to present my postdoctoral research on a vector-borne pathogen that's pretty prevalent here in the United States, Lyme disease. So the title of my talk is called Battling Borrelia. The pathogen that I'm talking about is called Borrelia burgdorferi, which is the causative agent of Lyme disease. Borrelia is a gram-negative spirochete. So this is an EM of the bacteria. You can see this very characteristic corkscrew shape. And that's because the flagella, as opposed to other bacteria where the flagella is outside to promote motility, Borrelia has an endoflagella. So the whole flagellar structure is actually sandwiched between the outer and the inner membrane. And the tension and the torque of the flagella gives it that corkscrew shape and also promotes the corkscrew-like swimming motion of this pathogen. So it's a very, very nice pathogen to look at. It's very active, but it also has really severe consequences if you happen to be infected. Borrelia is not a free-living bacteria. It exists in a natural reservoir. inside small rodents or birds and up to larger mammals like deer. And they are maintained and shuttled through across these different natural reservoirs through the feeding activity of its vector, the black-legged tick, Ixodes scapularis. So this is a picture from the CDC website showing the different life stages of Ixodes. The larval stage is smaller than a poppy seed that you might have on your favorite bagel. So it's very, very hard to see. So on occasion, usually through human activity, depending on where we like to do our hiking or where we even live, as the areas that we develop for habitation start to encroach on natural environments, you then end up encountering ticks that carry Borrelia, and that's when you can be inoculated or introduced to the pathogen. So we are actually not the preferred hosts of Borrelia. We are dead-end hosts. So from the point of view of the pathogen, they don't want to be in us. They want to be in these natural reservoirs where they can just circulate. However, when pets or humans are fed on by infected ticks, you can get presentations of Lyme disease. So the acute presentation of Lyme disease will include fever, pains, but the characteristic phenotype or characteristic symptom that you get in 70% of cases is this bullseye rash at the feeding site. If left untreated, the bacteria at the feeding site will then disseminate throughout the body through the bloodstream and will start affecting different tissues, and this is where you get into the more chronic stage of Lyme disease, where you might develop arthritis, which is the most common presentation, but you can also get neuroborreliosis, so affecting the CNS as well as the muscles around the heart. So in terms of how common this disease is, so it is still the most common vector-borne disease in the United States. The highlighted states in this map show you states of high incidence, and the individual dots show you confirmed cases. And you can see concentration of cases in the Northeast as well as the upper Midwest, and very sparse elsewhere in the United States. The name Lyme disease is named after the town where the disease was first characterized. So Lyme, Connecticut, there was an outbreak of arthritis in children, which is very atypical, but like a very entrepreneurial nurse was able to identify this pattern and then was able to sort of establish that it's an infection and not just a random event. So that they named the disease after the town that was described. Over the last 15 years and more, the incidence has been steadily increasing. So this is telling us that the current strategies that we have to combat this disease is really not working. And part of the reason for that is we don't really understand how Borrelia promotes infection. So we don't know the molecular mechanisms of how this bacteria, in conjunction with the influence of the tick, promotes initial colonization of either the natural reservoir or of us. So I sort of used my genetic training, and we did a knockout mutagenesis screen. Borrelia, like other bacteria, at least are very easy to genetically manipulate, so we're able to make a library of different knockout mutants. And we have a mouse model of infection in the lab, so we can introduce the mutant Borrelia into mice by needle inoculation and look at its ability to colonize. So we're looking for genes that inhibit or block its infectivity. And we define infectivity or colonization by its ability to colonize distal tissues. So that would be the joint, the skin, the heart. So we inoculate at day 0 and we look at day 21 at the distal tissues to see if the mutants are there or not. To cut the long story short, we were able to find one new gene in Borrelia that promotes infectivity. And the gene is uncharacterized until we found it, and so it just has a letter designator, BBK13. And the phenotype for this mutant is as follows. So we inoculate our mouse with either wild-type Borrelia, the mutant Delta BBK13, as well as complemented version of the mutant. So we're restoring the wild-type copy in the mutant background. And we look at day 21 in different tissues. Our initial screen involved a very quick but semi-quantitative way to assess whether we have viable spirochetes at the distal tissues. What we do is we dissect out different tissues, put it in bacterial growth media, let that incubate, and just inspect visually if we see these free-swimming spirochetes. And we have a rubric of how to score the density. And so what you can see that wild type is very good at infecting distal tissue, so it's getting the highest score of 3. The mutant, you can see, has 0s and 1s, so it's a lot more. It's still getting to some of the tissues, but a lot more reduced. And the gold standard in genetics to assign a phenotype to a gene is that the complement restores wild type levels. So we did this semi-quantitative method during the screen because it was not labor intensive, but we needed to validate this to make sure. So this is where we're actually quantifying the amount of spirochetes in the distal tissue. And to do that, we extract total DNA from the distal tissue, so both the mouse DNA as well as the Borrelia DNA. And we use qPCR to enumerate genome copies. So how many genomes of Borrelia do we find normalized to the mouse host genome? And you can see here the squares represent the genome copies of the mutant. Delta BBK13 is always low compared to wild type, and that's true over time also. So in this case, we sampled at multiple time points. So from 2 weeks to 4 weeks post inoculation, it's always lower than wild type. So not only is it low at one point, it doesn't really recover. So it's not just it's lagging, it's really reduced over time. And that's true across different tissues, and the dashed line represents the complemented mutant and shows that it's going back up to wild type levels. So again, confirming the role of BBK13 in infecting distal tissues. So this was great. Discovering like a new gene for infectivity is always a good thing, but it really doesn't tell us much about what is happening between day 0 and day 14. So we know that it's already low by day 14, but we wanted to know at what stage in the infection is the deficiency happening. So, I went ahead a little bit. So this is the same assay where we try to increase the inoculum dose to see if we can recover. We can track it. Is this just a matter of we're not just getting enough bacteria in there? It doesn't really matter whether if you go up 3 orders of magnitude more of the bacteria, you're still inhibited. So the requirement. So this just emphasizes the requirement for this gene in an infectivity. So going back to tracing sort of like where during infection the deficiency happens. So as I said, bacteria is introduced in the skin. It disseminates through the blood to reach these other distal tissues. So when we look one step back and look in the blood, we can collect blood from inoculated mice and plate it on culture media, and we just count colony forming units of Borrelia. So what this shows is that it's actually really interesting because you can see a peak bacteremia in the blood around day 6 to 7, and then it goes down. So there's this very brief window where the spirochetes are actively present in the blood, potentially reaching different tissues, and then it goes away. So the peak of that happens around day 6 or day 7. The Delta BBK13 Borrelia has reduced numbers in the blood, and the complement, the dashed line, restores it. So it's deficient already in the distal tissue. It's already, and then we're seeing this data shows it's already deficient at the dissemination step. So we move one step back. At at the needle inoculation site. So we're doing kinetics. We're trying to enumerate copy number at the skin. So this is at where we inject the bacteria. So what's very interesting here is that we've hardly detected between, at day 0, so hour 0, so immediately after inoculation up to day 3. And what we think is happening is there's a massive clearance event of bacteria. So not all of the bacteria survive, but enough do. And then from day 5 to 7, then we get this massive increase in genome copy numbers. And I'll just mention here, we've never, so compared to the distal tissue and then the blood, the skin inoculation site had the most, we've never seen numbers as high as what we found at the skin inoculation site. So what we think is happening is that as when Borrelia goes in the skin, there's a lag phase, and then it undergoes this rapid expansion in population. And we see that in the wild type Borrelia, but the mutant is not expanding as much, whereas the complement goes the same. So what we think now, what we know, so even though we discovered this initially as a defect in late colonization, the gene actually acts at the earliest steps of infection, at the skin. And what this actually told us more about the biology of Borrelia and infection is that you do need this rapid expansion in the skin. Do you need this many spirochetes in the skin? Probably not, but at the skin. But what it does is it drives the numbers of spirochetes so that there's enough spirochetes that move into the bloodstream, and then there's enough of the spirochetes that land on the distal tissues to establish enough colonization. If you were in the natural reservoir, so that there's enough Borrelia on the skin so that the next tick that feeds on you can acquire it and continue the enzootic cycle. But if you're a human or a pet, if there's enough Borrelia there, you might start manifesting the symptoms of Lyme disease. So what we've really uncovered here is the importance of the very, very early events in infection within the first week at the skin. Because if you can reduce the numbers at the skin, we already have data that you can reduce the numbers all the way out to the distal tissues, which potentially can either reduce or prevent any symptoms of Lyme disease if you were able to block it at the skin. And what's sort of interesting here in the field of Borrelia, so I wasn't trained in the Borrelia field, so I was coming into it as an outsider. A lot of people have focused on the pathology, so they're looking always at the distal tissues because the disease has joint manifestations in humans or heart manifestations. And they've sort of ignored the middle stages. And then we find, these genes that have roles early on, and they're like, oh yeah, that's true. We should have been looking at that earlier. So what I've been able to contribute to the field is to refocus everyone to what's happening in the early infection, because it has impacts all the way out. Right, so as a vector-borne pathogen, we don't have needles. Well, I mean, there are needles in nature, but naturally what happens is a tick will feed and transmit. Not a convenient needle that you can control the dose of. So, and we know that the tick saliva and the tick feeding activity it influences the immune profile of the feeding site. So what we can do in the lab is to actually recapitulate the whole natural tick acquisition and transmission, where we infect mice by needle. So these mice will now have Borrelia. We can put uninfected tick larvae on it and allow it to acquire Borrelia, and then ask whether so now we have infected larvae and see whether they can transmit. So what we find is that in mice that are infected with mutant BBK13, ticks can't acquire. So if you block the activity of BBK13, then here's one way for you to really block the enzootic cycle. The other thing this is really sort of where I ended, where, So the ticks that we had here were freely feeding, so they were anywhere they want to go in the mouse. It's hard to then ask what's happening at the skin because the ticks can feed anywhere. What I was able to develop in the lab is a tick capsule feeding method where I can define where the tick feeds. So now I have the ability to do early infection kinetics using a tick-transmitted infection model because I know where they're feeding and I can recover that feeding site at any time point. And what we see, it recapitulates the needle infection model. We do get this expansion event in the skin also, but at a much later, everything takes longer when delivered by ticks. Instead of peaking at 6 to 7, it's peaking around 8 to 10. But we do see this up and down pattern in this model. Just really quickly here, if we have the tick deliver the delta BBK13 mutant, interestingly, the tick is actually able to bypass the phenotype in the skin. So the BBK13 mutant actually replicates as much as the wild type if it's delivered by the tick. But if you look at the distal tissues, it's still low there. So somewhere between its skin and the distal tissues, there's still a requirement for BBK13. I have data that I'm not going to show, and we think it's immune related. So we did a bunch of experiments on immunocompromised mice backgrounds, and we're able to show that that potentially is where BBK13 function and host function are overlapping. But it's still a very open question. So in conclusion, I showed that I discovered a new gene, a new infectivity gene in Borrelia that promotes colonization of distal tissues. I showed that if you disrupt this gene, you can block the enzootic cycle and potentially reduce pathology of Lyme disease. I brought focus back to this dramatic spirochete expansion in the skin during early infection, which drives the rest of the disseminated phase of infection. The skin is a very promising therapeutic location because everything's still localized and it's very accessible to treatment or any manipulation compared to once it's disseminated out to these deeper tissues. And it's an important but poorly understood phase of Borrelia infection. And lastly, this development of the tick capsule feeding method captures the interplay of the vector pathogen host triad. And now there's technology that's been developed fairly recently that allows you to genetically manipulate the ticks through CRISPR-Cas9. So you can now really manipulate all three components and really start to dissect the molecular mechanisms of spirochete infection with the influence of the natural tick vector. So with that, I'd just like to acknowledge my postdoctoral mentor, Dr. Molly Jewett, and members of the lab who contributed to the work that I presented, funding sources from the NIH and a private foundation called the Global Lyme Alliance, which supported 3 years of my postdoctoral work. Thank you for your attention, and I'll take any questions.
Hi, I was just wondering if this and two, as an application, have you looked into what can
So the sad part is, so this is I knew this was going to ask, but I didn't put it in there. We don't know what BBK13 does. The protein that it encodes has this, it's really just one domain. It's called a simple domain, and it's a transmembrane. There's a transmembrane protein here. The closest homolog that we found was actually not in lepto, but in Brucella abortus. There's a Brucella simple protein BP26, and someone has gone and crystallized the structure because this is this Brucella is of agricultural concern. So they wanted to know, and this is actually one of the main immunogens for the vaccines against Brucella. So they wanted to know what the function of it is. So the structure in Brucella looks like this. Assembles into this 14-mer pore-like structure. There's no real pore, but for most purposes, from everything else, it looks like a pore. And you have the outer membrane and then the inner membrane on either side, so it sits in the periplastic membrane. We don't know what people in the lab have tried some biochemical approaches to We know that it oligomerizes like this protein. We don't know whether it forms the same structure. We know it's membrane enriched in Borrelia, so it's in the same location. What I think is happening, the outer membrane of Borrelia is very loose. It tends to slough off. So it's actually a lot of lipoproteins instead of your typical outer membrane components. So what I feel is happening is if you take BBK13 out, the outer membrane is a little bit not intact, and it might be affecting the amount of other surface proteins that are important for mediating, interactions with the host. There's a few candidates that's been described before, some outer surface proteins. So I think so. I've left. I've stopped working on this project by then, but I think the direction was whether see whether these known proteins have changed in levels or localization. But we don't know what it is. Is the short answer. Yes.
There's a request from the Zoom audience to please repeat the question, if possible.
Oh,
Yeah.
this one?
Because we have.
Oh, I see. Sorry, the question was whether we know what BBK13 does, and can we, if we know what it does, can we use existing drugs to target it, in the treatment of Borrelia? So the answer is we don't know what it does.
Any more questions?
Yes.
Giselle?
So is it also a biofilm format?
So the question was, does it form biofilms? We haven't seen it form biofilms. It's actually quite a controversial idea in the field. I don't think it's completely excluded, but the field doesn't think so. It doesn't exist in high enough numbers in the host. So it's there, but it's very sparse. And they don't really clump up in the same way you would need in a biofilm. But there's enough people there who think that it can form, maybe not a formal biofilm, but some sort of aggregation. Because we do see that in culture, but whether that exists in the actual, in vivo environment, we don't know.
Because I was thinking about it, the antibiotics, some antibiotics work on it, but there is a persistent form of it.
Yes.
I'm just curious whether it's because,
Right. Yeah. So this is where, so the field is sort of split here. So it's called post-treatment Lyme disease syndrome. I think they formally acknowledged that this does exist. The bacteria is very susceptible to antibiotics. It doesn't really have any, antibacterial resistance, but in patients, after treatment, in confirmed patients, they do notice that the symptoms don't go away. So it could be arthritis, but it could also be neurological, which is the more insidious one, because how do you don't know how to treat it. So they're exploring that. I know there are people that's funded by the Global Lyme Alliance who are looking at natural products also to try and see if that can address the persisters. I think there's no good model for the persistent model. I think that's what's missing. But really the field needs to invest, and it's investing in diagnostics as well as vaccines, because the bacteria is not really resistant at all. So if you can treat it early, either by knowing that you were infected and you get early treatment, or a vaccine which confers passive treatment, then that's probably the best across the board. What's actually interesting is scientists from University of Tennessee did pass a vaccine, but their approach is actually very ingenious. Instead of vaccinating the humans, they vaccinate the natural reservoir. So they're vaccinating wild mice, trying to reduce the load of spirochetes in nature, and potentially, maybe we don't get it as much. They've had much more success of clearing that through the regulatory bodies than human vaccines. So.
One more question. Yeah.
I was wondering if you have any idea as to what drives the apparent inhibition of BBK existence and persistence
Yeah.
of disease to the northeast and a little bit to the west of the north?
Yeah, so that's a good point. It's really so there's a lot of ticks that could carry bacteria or other spirochetes. And it's really where they're endemic. So it's really where these ticks are endemic and where their natural hosts are. Because you have tick-borne diseases in Texas, you have tick-borne diseases out in the West Coast and in the mountain region, but it's not Lyme disease. It's another bacteria and another tick. So they all co-evolve. So the ticks and the pathogen, and they're sort of like their natural hosts of all sort of co. And we think that's why they're sort of like localized the way they are.
Okay, let's, thank you very much. We have one more speaker. Hang on. Okay, who has
And I
it?
would actually prefer to.
Is it here?
So,
Sure, sure. Okay.
it's PowerPoint. Okay.
Everybody stand up and stretch. While we, yeah, I'll just read the bio notes. Everybody. You don't want to stand Okay, our next speaker is Dr. Frank Simmen, PhD.
Excuse me.
He's a professor
Sorry
at
about
the
that.
Department of Physiology and Cell
Just give it
Biology,
1 second.
faculty member of the Cancer Biology Program at Winthrop, Rockefeller Cancer Institute, University of Arkansas for Medical Sciences. He received his PhD in University of Hawaii in biochemistry. His postdoc at Baylor College of Medicine in cell and molecular biology. So his laboratory works in the areas of molecular endocrinology, molecular nutrition, and animal models of disease. And research in his laboratory is focused on, number one, molecular mechanisms of action of dietary factors and obesity in colon cancer. Number two, molecular physiology of metabolic enzyme 1, NE1, and the characterization of Krüppel-like factors in normal and pathophysiology.
There you go.
Dr. Simmen has served on several editorial boards of several scientific journals, including endocrinology, scientific
Projector
reports,
monitor is to your right.
cancer,
So if you move the
and
cursor all the way to the
He's
right,
also an active
it will show up here on
contributor
the.
to graduate
So it's like your
school and
second
medical
one.
school education and mentoring. Co-directs a nutrition model for second-year medical students. Here's Dr. Frank Simmen.
Thank you, Liza. Okay, it's very nice to be here. This is my first PASE meeting. I'm sure it's not going to be my last. It's been quite enjoyable so far. So I'm up here today because it was kind of a last minute request to Rosie or myself if one of us could, give a short talk in the immunology section. And after a bit of arm twisting, I ended up being the one up here. So what I've done is to now I'm actually not, a disclaimer up front, I'm not a card-carrying immunologist by any means. But what I've done is to go into some of the work that we've done to see if we can find something with an immunological slant that maybe fit a bit better with this section. So that's what this title is about. So this is sort of a couple of short vignettes on where we've gotten into cytokines and colorectal cancer in mouse models. Okay. Let's see. Where's the, how do I advance this? Oh, over here. Okay. All right, so we're not going to focus too much on this, but so my lab over the last 25, 30 years has focused on GI and liver pathology and normal physiology. And this is a recent summary from a paper that gets published every year. And the rankings have not changed. So colorectal cancer is still third highest ranked cancer for men and women in the US in both incidence as well as lethality. So it's clearly a very significant cancer. We are making headway on cures, chemotherapies, in particular a subset of colorectal cancers, which are defined as hyper unstable, are responding well to these checkpoint immune inhibitors. Anyway, it's clearly a disease that needs some new answers. And then you can also see down here that liver and the biliary tract cancers are also very lethal. And so the second vignette I'm going to show you deals a little bit with that aspect. So what are KLF transcription factors? So this talk is focused on Krüppel-like factor 9, or KLF9. And this is a class of 17 highly conserved transcription factors, KLF9 being the 9th discovered member of that group of factors. They're nuclear proteins. They have defined cis elements or DNA binding elements over here. The DNA binding aspect of the protein is actually comprised of 3 contiguous zinc finger DNA binding domains, analogous to the Drosophila protein Krüppel. So these factors are either activators or repressors of target genes. They remodel chromatin in cells, and KLF9 in particular has been implicated in stem cell renewal, cell death, cell proliferation, cell differentiation, as well as cell migration. So how did we get into KLF9? Well, I'm married to Dr. Rosalia Simmen. She actually put her name on the map in the late 80s, mid to late 80s, and the 90s, working on uterine embryo interactions and uterine genes that mediate successful pregnancy establishment. And one of these her lab found was this novel protein, Krüppel-like factor 9, in the maternal uterine endometrium, which seemed to interact with progesterone receptors, estrogen receptors, and had a key role in pregnancy establishment. So we talk a lot of science at night, and we decided, well, at some point, let's have a look at cancers with this protein. And we actually, in an early study, collaborative study, 2008, and then a follow-up study in 2011, we were the first groups to show that this transcription factor had reduced gene expression and protein expression in cancer, and this was human endometrial cancers. So typically, if you work in any kind of area and you find diminished gene expression for something, your first thought is maybe it's a tumor suppressor, so let's look further at it. And then some follow-up studies from other labs have supported this idea. Some of these are shown here. And then if you go into Oncomine or any the TCGA databases, other of these, you can easily find that this is true for human colorectal cancers as well. So this is the expression levels in normal colon mucosa, rectal mucosa, and in the tumors. So clearly there's significantly diminished expression of KLF9 in the tumor compared to the normal. So when we got into this, we sort of took a simple hypothesis that it was a tumor suppressor, and maybe it worked both locally as well as in an integrated fashion. So we've pursued both of these angles through the years with a focus locally using mouse models, and then in an integrative fashion, focusing on liver and adipose, since KLF9 is also highly expressed in those two tissues. In fact, it was actually first identified in rat liver, and then, of course, Rosie's studies in the uterus. Now, when we developed this hypothesis, we were also intrigued by the positive associations of non-alcoholic fatty liver disease, or NAFLD, body mass index with heightened colorectal cancer risk. So again, that gave us the direction for maybe an integrative pathway to pursue. So we used, in our studies, we used a well-characterized model for colorectal cancer, the APC-Min mouse, and so I'm not going to belabor you with the details of this, except to say that mice that are haploinsufficient for the APC tumor suppressor develop a fair number of adenomas in both their colons and their small intestines. So what we did was to intercross the KLF9 null alleles into this backbone in mice. Okay, and this is actually a shout out to Leah. I don't see her here, but we actually did our studies in both male and female mice. Okay, so we took the time to do that, and we actually did find some differences, subtle but still differences. Anyway, if you look at this data, you can see even removal of one allele of KLF9, we're getting more in the colon. We're getting more adenoma numbers. Okay, if we take out both alleles, it's about the same. So this is kind of pretty good evidence in both males, and in the females, it's the total null where we see a significant induction in the colons of adenoma numbers, suggesting yes, maybe it is a tumor suppressor. In the males, they're more susceptible to loss of one as opposed to the females. Okay, so that's a sexual dimorphic difference. We then took a look at adenoma size, and you can see again here, yes, in the female mice, or in the male mice, I'm sorry, excuse me, we not only get more adenomas, they're bigger, okay? In the females, we get more adenomas, but it's the smaller size class of adenomas that actually get bigger. So another sexual dimorphism in response to our knockouts. Now, unlike the colon, so the colon is a small region of the mouse GI tract, but that's analogous to the human colorectal rectum. The small intestines in this mouse model develop a fair number of adenomas, but absence of KLF9 had no effects on those numbers. So it seemed to be a colon-specific tumor suppressor. And I forgot to mention at the outset, when we do our adenoma monitoring, these mice are 4 months of age. So we've done the intercrosses. It takes about 4 months to get significant numbers of adenomas that you can observe morphologically. We then went back and did another study where we generated mice, the same backgrounds, except we sacked them 2 months earlier, so prior to overt adenoma formation, but we feel like we've set up the colon mucosa in some way to favor development of adenomas. And then we did microarray analyses, and one of the most interesting things to come up was we found an array of interferon inducible genes that were in turn induced in the colon mucosas of both heterozygous and homozygous null APC-Min mice. So all of these genes are immune-related genes. They're well characterized in terms of their interferon inducibility. They'd never been linked before to Krüppel-like factor 9. And they actually are kind of confusing if you think about some of them in terms of what's known in colorectal cancer in humans. But we'll get to that in a few minutes. So we identified this module of interferon-inducible genes. We then, using human colorectal cancer cells and knockdown approaches, we were able to recapitulate those results. So here we've simply done siRNA knockdowns of KLF9 and real-time PCR, and you can see we get dramatic inductions of virtually all of these genes. So it's kind of, the in vitro is mimicking the in vivo, and perhaps as importantly, these are epithelial cells, epithelial adenoma carcinoma cells, which are not always considered to be interferon responsive in vivo if you're looking at a colorectal tumor. All right, so this is more evidence showing again that if you, in this in vitro experiment, we've simply added recombinant interferon beta to these same HT29 human colorectal cancer cells in the presence and absence of knockdown of KLF9. And again, we're seeing knockdown of KLF9 actually augments interferon inducibility of these targets in vitro. Okay, so focusing a little bit on a couple of these targets, this is one of them, ISG15. We keyed in on this because it's homologous to ubiquitin. There's a parallel process to ubiquitolination involving ISG15, and it's known to be overexpressed in a number of tumors. CD133 positive colorectal cancer stem cells make more ISG15. More importantly, it's chemotactic. It promotes epithelial mesenchymal transition, etc. So it seemed like it's a good one for maybe knocking out, or loss of KLF9 induces ISG15. That maybe promotes tumor formation in our model. It also has a significant immune component to it here, so it's thought, there are some other papers which have suggested it actually is a secreted cytokine as well as functioning as an intracellular ISG-alation substrate. Okay, and the secreted cytokine behavior, one of the things it can do is actually remodel macrophage phenotype to an M2-like phenotype, which we consider to be maybe tumor promoting. Okay, so anyway, we took some of this and we showed that it's actually anti-apoptotic with human colorectal cancer cells in vitro. So that also fits with it being a tumor promoter. Another target we looked at was CXCL9. Since this was induced by knockout of KLF9 in our systems, and this again, like ISG15, is implicated in multiple malignancies. It can also induce cell migration, and it has a key role in T cell trafficking, okay? So T cells are important in terms of tumor response, tumor growth, whether the tumor is immune suppressed or immune privileged, okay? So T cells are very important in that aspect of it. So taking a look at this chemokine, just like with ISG15, if we used recombinant CXCL9, we also induce proliferation of human colorectal cancer cells. So we kind of built a story then that KLF9 is having semi-global effects. It's a repressor. If you get rid of it, it's inducing immune-related genes, which some of which are at least tumor promoting in the models that we've looked at. A third target that we found, which was, at the time, we didn't realize it was so significant, this molecule here, PDL1. So I'm sure you've all heard a lot about PD1, PDL1, okay? And these proteins are involved then in T cell interactions with tumor cells and whether the T cells become cytolytic T cells and kill the tumors. Anyway, knockdown of KLF9 actually led to an induction of PD-L1 as well. So presumably an imbalance in that then maybe led to some sort of altered immune milieu, if you will, promoting tumor formation. Now this is some data just that I took, I queried off of a publicly available single-cell RNA-seq database. These are human colorectal tumors, and we're looking at, on the left, sort of a two-dimensional representation of different cell types and the expression levels of KLF9 in this case. The cell types are clustered according to similarities in gene expression. You can see then, now this has nothing to do with, we have not manipulated KLF9 in this system. This is just human colorectal cancers looking at KLF9 expression. You can see that there's still significant KLF9 expression in many of the immune cell types in these tumors. Okay, the epithelial cells in here are relatively low in expression of KLF9, fitting with the data from Oncomine and TCGA, where tumors have less KLF9 than normal human colon. Now this makes it even more complex when you're going down the road. If you want to build on what we've done with the mice and our targets, I just put this together to illustrate that it's not going to be it's not going to be so straightforward as we're thinking. KLF9 induces or suppresses ISG15 and CXCL9 and PDL1, because while there is some overlap in expression of KLF9 with these targets in some cell types, there's not in others. And you can see then that, for example, ISG15 is actually rather ubiquitously expressed in the tumor, even in the adenocarcinoma cells, whereas CXCL9 is mostly in the myeloid cell lineage over here. Okay, so anyway, the point of these two slides is simply that single-cell RNA-seq is giving us a way now of looking at different cell types within a tumor and how they might interact. And in this case, it's actually implicating KLF9 in a variety of immune cells in the tumors. And I think that's pretty interesting and probably points to why we should study this protein further. Quick aside then, a troubling trend, you're probably aware of the fact that we used to consider colorectal cancer a disease of old people, but it's actually moving to the younger generations. It's mostly the rectal cancers that we're seeing more onset of in younger people. And some of us think maybe that's because of the increase in adiposity, the obesity epidemic that's been going on for the last couple of years. And so that got us into this second aspect, the integrative aspect of our work. So again, in this case, we're not using the APC-Min mouse. We're simply making mice fat by feeding them high-fat diets. And then we're going to look at liver phenotype. And I'm just going to show you a very small bit of data from this study. So again, we did males and females, okay? Put them on high-fat diets for 12 weeks, sacked the animals, obtained livers, did a variety of gene expression and histochemical assays. And I'm just focusing here because it's an immunology session. We got some rather dramatic inductions with immune-related genes, in particular IL-6, TNF-alpha, and interferon gamma, especially in the male livers, in the females, IL-6 as well. So to wrap it up, IL-6 is well known to be a promoter of liver cancers in humans. And so we have a good fit then with KLF9, maybe suppressing liver tumors by virtue of suppressing local and systemic IL-6 production. We also did a little bit with oxidative stress real quick. If we have an animal that's null for KLF9, its livers are characterized by markedly enhanced oxidative stress. So in these two slides here, we're just monitoring biomarkers of oxidative stress. So in this case, ratio of reduced glutathione to the oxidized form as a measure of reactive oxygen species. You can see a significant decline in that, so indicating KLF9 is suppressing oxidative stress or in some way countering that in the livers of high-fat fed animals. Looking at another redox couple, in this case, we're measuring cysteine conversion to cystine as a indirect measure of reactive oxygen species. I forgot to mention, interestingly, we get the same results in both the serum, the livers, and the corresponding sera from these mice. So livers, of course, filter portal blood, and we think they might have systemic effects. This mechanism might have systemic effects via the filtering of portal circulation. Okay, so in conclusion, 2 vignettes supporting the idea that KLF is probably a suppressor of colorectal and liver cancers. At least in the colorectum, we think there's part of this is this interesting suppression of a subset of interferon-inducible genes. In the liver, it seems to maintain an anti-inflammatory state and counters liver and systemic oxidative stress. And the hepatic IL-6 effect, the linkage with that is interesting both from local as well as systemic mechanisms of action as well, since IL-6 also promotes tumors in breast and colorectum and other tissues. Lastly, just a shout out to some of the people who've done the work and the funding sources. I would be remiss if I didn't mention Rosie, and her name is actually a little bit bigger font than everybody else. But I want to point out, it's probably hard to see, but this individual right here, Dr. John Mark P. Pabona, and then, oh, I lost, what did I do? Can I get this slide back? Okay. And then this individual here, Dr. Maria Teresa Montales, both were postdocs in Rosie's lab. They both did their MD training, University of the Philippines Medical School, came to UAMS, did their post. They're both now practicing internal medicine docs in Little Rock, Arkansas. Thank you. Yes, June.
I was able to appreciate your early slide on the association.
Right. Yes, it's so like in colorectal cancer, liver cancer, glioblastomas have markedly reduced expression of KLF9. It seems to work in that cancer through repressing the Notch pathway and Notch signaling. It's also, the suppression of KLF9 seems to trigger a stem cell phenotype in the glioblastomas. So there's pretty good evidence it's a suppressor in that context. Yes?
I thought the single cell RNA data was very interesting. I was wondering if you also look at the protein levels and if they correlated with whatever you saw with the mRNA models, and what we're finding out from our work is that
yeah, that's a great point. And no, I mean, that data was not our data. The Broad Institute, they published a paper in Cell a couple of years ago. I just quickly, when I knew I was doing this, I figured I needed some immunology. So I don't know. I mean, there are databases. We need to look at those. And that's a great point, though. But I would argue that in many of our studies of the last 35 plus years, so many people will say there's discordance of mRNA and protein. Many of our situations, we've done both and we find concordance. So I'm kind of in the middle on that, but any other questions?
There are no other questions. Let's thank all our three speakers, Dr. Simmen, Dr. Simmen, Dr. Tigno-Aranjuez, and other doctors. We are also giving them certificates of appreciation. They are, like I gratefully acknowledging their participation in imparting their valuable knowledge, insights as an oral speaker here in the 2023 APAMS with our theme, reimagining the training of 21st century STEAM workforce is the cell of today, June 29, sorry. And their certificates are signed by Dr. Mario Santo Domingo, Dr. Justine Tigno, the co-conveners. Thank you very much.
Let's have a 5-minute break. There are snacks outside. I'll let our next speaker know. Dr. Gomez, we'll have a 5-minute break. Okay, thank you.
Yeah, so that I can show. Let's go. No, this one. No. Oh. Oh my goodness. Oh my God. I'm embarrassed. Take that out.
You then look at that, it's just the cells, and then the environment also just the seal. You don't need to resolve the blood, and then once the
Trying to see if you can just give this to the young or apart from those who.
So,
We have a list some other people. One, two, from
Well, because we stayed in Charlotte, that's where. That's why it's. I think it's still fast, even though the traffic is still fast. Even in the express, yes. As long as I have like 3 noise using from, because if it's like smooth jazz or something, it depends on how energetic. If you go into Charlotte, there's no traffic on the ads, it's also, it's long.
For the government.
Okay, we'll start.
Okay, so good afternoon, everyone. From the heavy immunology topic, we now move on to engineering and technology applications.
Okay, we're starting already. That's intellectual.
Okay.
Excuse me, we're starting already.
All right, so I would like to introduce the first speaker, Dr. Ronald Gomez. And he is a professor and associate chair for undergraduate education of the Department of Electrical and Computer Engineering at the University of Maryland, College Park. And he obtained his PhD in condensed matter physics from the University of the same university. MS degree from Wayne State University in experimental atomic physics, and bachelor's degree from the University of the Philippines. Mel Gomez co-authored more than 100 scientific publications, including several book chapters and 3 US patents in the general area of nanotechnology. So apart from research, he also teaches undergraduate and graduate courses in quantum theory, electromagnetic theory, and magnetism, electronic circuits, and engineering design. As associate chair, he has implemented significant changes in the electrical engineering and CPE programs to modernize the curricula to better prepare students for jobs in the industry and graduate degrees. So he is a proponent of hands-on education and has spearheaded the development of several training laboratories in the department. He is also leading an effort to develop electronic engineering programs or electrical engineering in Southern Maryland and at the universities at Shady Grove. Let us all welcome Dr. Ronald Gomes.
Hello, hello everyone. Can you hear me?
Yes.
Okay, I wanted to share my slide, and I was wondering if I could do that. If someone would allow me to do it. Hang on. So I am currently, okay, allow open system preferences, Zoom. Okay, okay. So, okay.
You are a co-host, so you can share.
Okay. Grant access to Zoom. Wait a minute. Zoom. Privacy. Give me 1 sec, okay? Because this is a little bit. Okay. Okay, can you see my screen now? Not yet. Okay. Okay, just a sec. Okay. I seem to have some difficulty trying to communicate, trying to open up the. Okay, hang on. Okay. Later. Okay, now can you see my slides now? Not yet. Let's see. One more. Okay. Okay. Aha. Okay, where is my slide? This one. Okay. Can you see that? Can you see my screen now? I hope. Okay, can you see it?
Yes.
Okay, thank you. So, okay, so that was 2 minutes. So I would like to give you a little bit of a background on this new program that we had just developed here at the University of Maryland. This is the 3rd year. We started this program in fall of 2020. And so we now have graduated 2 cohorts. And this is the Bachelor of Science in Cyber Physical Systems Engineering. So let me start with the definition of terms. The first one is, what is the meaning of basically cyber physical systems? A lot of people confuse cyber with cybersecurity and so forth and so on. Well, cybersecurity is a small part of the whole universe of what we define as a cyber physical system. Fundamentally, cyber means it's anything that has a computing capability and communications capability, iPhone, laptop, etc. That would all be cyber. Now, physical systems, of course, is anything related to, well, the rest of the world, mechanical, chemical, electrical, optical, quantum properties. And so the idea behind a major like this, it's necessarily multidisciplinary. We would like to put them in a single undergraduate major in the engineering department. Okay, so now let me just tell you a little bit about the advances of what we call technical laws that drive the cyber physical world. This is the world that we live in right now. And just to give a little bit of update on what technological progress we've been enjoying the last decade or so. We all, of course, probably know about Moore's Law. And that means that the fundamental building blocks of electronics, which is transistors, their density doubles every 6 months. There's Kumi's law, which says that computing efficiency doubles every 18 months. There's Gilder's law, the communication bandwidth. In other words, the amount of information that could be transferred from one system to another worldwide grows 3 times, a factor of 3 in 3 years. And there's the Schreider's law, which basically says that hard disk, the fundamental manner where we store information in a non-volatile way, doubles every 18 months. And finally, there's the Metcalfe law, which is economics law, which says that the value of a network goes as the quadratic or square of the number of nodes. Now, I could go through the history of this. Is a whole new talk altogether, but fundamentally, these 5 laws are the ones that drive the digital age as we know now. Well, what is the state of the art? I think it's very important to know what the state of the art is. And so this is kind of the latest that we have so far, probably about a year or so old. As far as the Moore's law, chip density, the amount of transistors that you have is now so high that you could have something like 5 billion transistors per square centimeter. And that's the latest Apple A11 microprocessor. Now think about that for a moment. The amount of about 1 centimeter square is about the size of your thumbnail. Imagine that you can stuff 5 billion transistors on that. That's a lot of components that you can put. And of course, there's a lot of sophistication included in it. What about computing efficiency? Well, there's the thing called FLAP, which is floating point calculation. Basically, it's a unit of calculation. So this unit basically says that you've got 10 to the 18th kilowatt flaps per kilowatt hour. What does that mean? That the amount of, well, kilowatt hour is probably what, I don't know, 20 pesos or probably 20 or so cents. That's the amount of energy you need to be able to compute 10 to the 18 times. So as far as we're concerned, computation seems very, very cheap, right? That is why we got blockchain technologies, Bitcoin mining and so forth and so on, which is very intensive, had become very popular because we now have this tremendous computing efficiency, rather inexpensively, if you might add, 10 to the 18 floating point calculations per kilowatt hour. What about global bandwidth? This is the amount of how fast your internet, not internet, but communications. This is now 1 terabyte per second. I'm not talking about Wi-Fi or 3G or even 5G. We're talking about optical communications, the one that link from continents to continents. These are all fiber optics. The important thing to note is that we are now at about 1,000 terabytes per second. That's the amount of information that can flow through the internet. That is why you can see me almost in real time, even though we're, I don't know, 600 miles away. Excuse me. As far as non-volatile, the amount of memory, how much does it cost, for example, to store data? Well, it's basically $20 per terabyte. Well, terabyte may not be so, it's not so small. It's probably the amount of, if you have a digital footprint, even though you probably use TikTok or whatever, all of those documents, let us assume that every human being, has some electronic footprint. About a person would probably have about 1 terabyte in one entire life, in 1 year. What does this mean? Think about it this way. Anytime you publish something on the web, anytime that you can, it's accessible through the web, you can assume that it is being stored somewhere. Why? Because it's only $20 per terabyte. And think about how much knowledge I can get by simply storing everybody's information. Remember, it's only $20 per year per person. And that's an awful lot. That's very cheap to actually be able to monitor the physical, the footprint of individuals. What about network value? What is economics? It basically says the number of nodes. 2 people talking, that'll be 1, 2 nodes. You've got 3 people, you got 3 nodes, and so on. Well, it scales. The value of it scales quadratically. to put it in perspective, let's look at Facebook. Facebook has about, I don't know, about 4 billion users, active users, nodes. If you multiply it, user square, that's 9 times 10 to the 18. That's 10 to the 18 units. Now imagine if you're just gonna penny for all of those, for every transaction, that's a lot of pennies. So that's the nature of why these big companies, Google, Facebook, etc., the ones that TikTok and so on, they all want to grow in terms of the number of nodes, because the number of nodes squares. And that is there's plenty of money to be made, and probably money to be lost as well. So that is the nature of our digital age. We are now way past. This has only been dreamed about 10 years ago. We are here. We are actually very, very capable as a civilization as far as technology is concerned. So then, what about? Well, we also have mathematical models. There's a big rise on artificial intelligence and computation. Of course, we all know about computer vision, natural language processing, the chat GPTs, neural networks, deep learning, quantum computation, and so forth. These are all advances that have been happening over the last 5 or so years. So now, what is the problem then? Well, okay, these are the common applications of embedded system of cyber physical systems. We all know about self-driving cars, whether we like it or not, whether we would actually trust Tesla to drive us around. That's a different story, but they're all present in self-driving cars, in drone swarms, electrical power grids, Industry 4.0, healthcare and medicine, biocomputation, security, entertainment, commerce and logistics, environment, agriculture. As a matter of fact, about everything that we do in the human civilization can be enhanced by cyber physical systems work. And that's the value that we bring to the society. Now, there's therefore a need for innovative curriculum. We have traditional electrical engineering, computer science. Now, I submit to you that those things are far too narrow. Those majors are far too narrow to be able to address the needs of the technology in the coming years. We're talking about in about a decade, maybe 2 decades from now, there's going to be a sentient computer that's probably running the world. But that's probably science fiction. The point I'm trying to make is all of our degrees right now, the computer science and computer engineering, are so limited. They're so focused, narrowly focused on their areas. So we need something a little bit broader. Perhaps we should relax the depth of certain things, but increase the breadth of it. And that's the reason for introducing this program. So now, so again, let me just, this is probably a little bit too boring, especially because of the previous sessions are all in RNAs and genes. This is a little bit probably not as interesting to some, but here's a comparison between computer science and computer engineering. That's the two closely allied fields. Okay? Computer science is about software engineering, programming languages, theory information. We have a little bit of computer networks, edge and cloud computing. The difference being edge computing is stuff that compute on the phone. Cloud is something where you use machine learning or artificial intelligence to do things like, for example, Siri. That would be something that's cloud-based computing. But then you also have software. Of course, they should know how to program discrete structures, algorithms, computer architecture, machine learning theory. That is all in the red. That would be computer science. Notice there's no hardware associated with it. People don't know electronics or whatever. But that's the area where computer engineering now shines. Computer engineering, we talk about what is known as analog and digital circuits. It's basically, you know, how do you build these things at the level of hardware? How do you solder things around and do this? So that is introduced in computer engineering curricula all over the world, pretty much. We have computer hardware. We have microprocessors. You know, how do you design a CPU or GPU? Those are all considered in computer engineering. At the same time, you also have specialty topics like signal processing, or how do you compress signals? How do you make sense of signals? How do you make interpretation of various visual models and so on? That's all in electrical engineering, in computer engineering. But what is needed is to integrate this. So we need information tech, we need IT, we need cloud computing, we need interface. We need to be able to have the user interface and the thing that goes beyond it. So what we need is networks, but we don't have that. And that's the reason why we are introducing, here is the cyber physical systems curriculum. All of the stuff that is, cyber physical systems here has this communications, gateway, and protocols, how two computers can talk to each other, IoT, system of systems, hardware interfaces, user experience, machine learning tools, robot operating systems that allow you to control all sorts of devices. And that is included in this. So it has it is common with computer science in the sense that we both talk about computer networks and edge and cloud computing. It has a great deal of common with computer engineering, which is analogs. So in other words, all of this thing, and of course, all of them share the fundamental engineering, essentially pillars in this field, which is coding, structures, algorithm, and computer architecture. So that is the nature of this program. So it essentially takes away some of the unnecessary. Well, it combines all of the necessary topics into one, in order to provide the student with a far more broad understanding of how the technology works, how to make the technology, and actually bring products to life. So then this is the curriculum. It is a lot of words, I know, but please bear with me. So here we begin with engineering foundations. Everybody who has done computer engineering, computer science, even electrical engineering will know a lot of these fundamentals, analog and digital circuits, processors, embedded systems, etc. And as we move higher in the curriculum, we see a lot more of the applications I talked to you about earlier, about robot operating systems and IoT applications, Internet of Things, by the way, and communications and protocols. So this is the structure of the curriculum that we are offering, and I should sa Tagalog magbubuhat ng sariling bangko. It is the first in the United States. This is currently being accredited in the ABET. We're in the process of ABET accreditation. And would you believe they have to create, ABET will create a new subfield specifically for the cyber physical systems engineering. So we are kind of trailblazing in this field, which I am very proud of, that we are doing it here at the University of Maryland. So how does it work? I mean, what is the unique feature of this program? Well, we admit students that already pass the general education requirements. In other words, we admit anyone with an associate's degree in any STEM field. Doesn't matter if it's physics, mathematics, mechanical engineering, chemical engineering, etc., or those with associate's degree, so that they are already past the general education requirements of a program. The other option is for students to enter the program from a four-year institution that already are finished with the general education requirements. You see, in the academic world, there is always this general education requirements that everybody, all universities would have to satisfy. That includes, English, plurality, cultures, humanities, and so forth and so on. Those are great courses, but we would like to get those students who already completed all of those so that we can then focus on the technical aspects that we would like to teach. That is why this is only open to those students who had already completed the general education requirements. I should also point out that another unique feature of this is that this course is taken not at main campus at College Park, because we simply don't have resources to do that. We are doing here at the universities at Shady Grove. This is a brand new facility. This was built in 2019, specifically to house the program such as ours. And so it's a lot of resources had gone into building not only the building but also the infrastructure that goes with it. So then that's the idea. But how do you enter the program? it's an engineering program, and so there are qualifications, just like with any kind of what is known as limited enrollment program. Not anybody can go in with that accepted. You have to meet certain requirements for very good reasons. You have to have solid, well, mathematics background, so we expect that our students would at least have a 3.0, B average in calculus 2, and maybe physics 1. Chemistry, a lot of students are struggling in chemistry, so it's okay to have barely passing. C- is fine in chemistry. Those are the 3 pillar math and science courses that are required. And we also do not want students to spend a lot of time getting good grades by repeating the course. So there's a 1 repeat policy. If you repeated calculus 1 to get the required calculus 2, for example, to get the required GPA, that's it. You cannot repeat physics, cannot repeat any other thing, because we do not want students to simply spend a lot of time spinning the wheels without really doing any progress. We know if a student would have to repeat 1 or 2 technical courses, then it's going to be a little bit of an uphill battle for them, which is why it's a little bit more, it's a little restrictive, but that's the way that we have to do in order to implement a successful program. And in addition to that, we also want some of our students to have some advanced mathematics. So differential equations, complex analysis, linear algebra. Those are those are classic advanced courses, that math courses. And it's not so much the mathematics foundation, but it's the mathematical maturity. I mean, how do you think mathematically? That is the reason why we require them to have advanced math courses. We also want students to have introduction to programming. It doesn't matter if it's C, Java, or whatever, Python. It doesn't matter, as long as students understand that there is a process, a thinking process in how you talk to computers, to programming. And finally, we would like our students to have engineering design, at least a freshman level engineering design. The idea of thinking of a product or thinking of a problem, thinking of the market, thinking of all of the particular solutions to it, that would be something that's essential to get into the program. Once we have them, we are going to offer them 60 credits. It's very, very short. Only 60 credits of very, very specialized courses. And here's a 4-year plan. I guess this is going to be very boring. I don't want you to read this in any kind of serious way, because I'll explain to you in the next slide. But essentially, the course is not so bad if you think about it. It's only 17 credits. The maximum credits per semester is just 17. The others are very low, 15 credits or so. So altogether, this is 62 credits that the students would take with us. And then they're going to be given the degree. So let me just very quickly mention to you the scaffolding of these courses. So the first, I got a little bit excited about using emojis. And so these are basically the courses. In the first semester, we have got analog electronics. I mean, analog electronics is like, how do you work with electricity? How do you work with signals? I mean, how do you make an FM receiver? How do you get these data? I mean, how do you get signals from the brain, for example? These are all electrical components, and so we need to teach our students the fundamentals of how to control, how to understand, and how to shape these electrical circuits. That's why we have analog circuits. We have digital circuits. People should know basic ideas of Boolean algebra, for example, ways to combine ones and zeros in clever ways. That is the reason for this foundation in digital circuits, discrete math, C programming, and introduction to IoT. IoT is the Internet of Things. In the introduction to IoT, it's basically we teach the students, how do you create, how do you use Amazon Web Services, for example, to control, something remotely in another region of the world? How do you turn on the light at home when you are in the Philippines, for example, and so forth and so on. So these are the key features that our students would have to learn in the first semester. In the second semester, it's a little deeper, microelectronics and sensors. We teach them about what is the signal transduction? How do you, for example, get signals? I mean, We talk about some medical field. I mean, how do you get blood pressure? How do you get the white blood, the saturation levels, for example, of blood? I'm not a doctor. I just know the principle behind how do you measure the supply of optically the supply of oxygen. These are all fundamentals, so that's something that we teach the students. And of course, computer organization, how do you arrange a computer? Well, how do you, there's a memory, there's a program stack, and so forth and so on. And of course, then there's algorithms. Algorithms is how do you process things so that it's the quickest way? Or what is this most straightforward way to solve a problem? It's basically algorithms. This is essential if you're going to deal with machine learning applications. And of course, we have to teach students networks and protocols because that's the essence. You have to know. I mean, every time you probably don't even care about it that much because every time you have a problem, you probably talk to your IT person. And so our students basically know this is the meaning of how you route it. There are so very, very, very standards that you have to obey. This is how you deal with it. So you can think of it as probably essential if you want to deal with cybersecurity, for instance. And of course, very, very important for everybody is probability and statistics. That's the first year. And then in the second year, it's a little bit more advanced. We have, of course, we need to teach our students how to write. A lot of our engineers are good in calculations, but probably not very good in writing and communications. That's an essential element of their education is writing. And then these are embedded systems. How do you create something that will function deep inside a machine, for example? You embed an intelligence, an intelligent device inside your computer. So I have an iPhone, for example. An iPhone might have a couple of hundred of these, maybe more, of these embedded systems that do specific functions. An example of an embedded system is, how do you adjust the focus of the camera? How do you adjust the lighting? How do you adjust the power, etc. Those are all brains. Those are all computer systems that monitor what's going on and tells the system this is what you should do. That's embedded system. Of course, in cars, you can think of so many things in cars, from the wiper blades to the braking, anti-lock braking system, to cruise control, to whatever. In airplanes, it's autopilot and so on. Those are tiny brains, which we call embedded systems. These are tiny computers that control things locally. We teach them that. And their operating systems, how do you do that? How do you schedule the timing? It's also so arcane at this point, and I apologize, but that's the essence of this degree. The most important thing here is the fourth semester, we got all of these things in orange. These are all elective. It depends on what the student might be very interested. They might be interested in advanced software for connected systems. For example, people want to work with Amazon Web Services, or people might be interested, for example, here in ROS, robot operating systems. They might be interested in unmanned aerial vehicles, for example, designs of robots. There would be something here. And of course, some people are interested in web-based applications. How do you program the front end, the one that faces the client, and how do you program the back end, what's going on beyond on the server side, and so forth and so on. FPGA is a modern tool, floating point gate array. That is essentially it's difficult, but it is the fastest way to bring a product into the market. That's basically it. So now, yeah.
Last 2 minutes, please.
2 minutes, okay, that's perfect. So what are the eligible jobs for our students? Well, they're expected to be good in software, understand the physical. So there's a whole lot of entry-level positions. Part of the reason why we offer this is because Maryland needs a lot of technical folks like these students, and so they can become software developers, software analysts, IoT architects, etc., so forth and so on. So there is no so they could fit in just about any field that requires competency in hardware and software and communication systems. So I just wanted to point out that the same faculty we have, I actually recruited faculty from College Park. We all teach here, and we teach both at College Park and here at USG. And the degree is not it's not a degree from the universities at Shady Grove or some other, but it is a degree from the University of Maryland, which of course is the flagship campus of the university system of Maryland. So they march together with everybody else when they graduate. So here's a building. This is actually where I'm sitting right now. It's a very nice building. It's dedicated laboratories. Here are some of the equipment, the specialized equipment that we have. I know that you probably don't. I'm just putting it because it's recorded in case someone wants to maybe begin a program like this. Then these are some of the components that would be essential to bring a program in the Philippines or some other parts of the country. So then finally, I would like to say a few things about our students. We have been quite fortunate because we said that we have to lower the barrier of education. So after the first year, we promised our students the first year with us that they would be given paid internships, which would be enough. They pay quite good money, so that it would be enough to pay for next year's education, the tuition and everything that they would need to go. And so it's essentially, just spend 1 year and then the rest you'll be taken care of. A lot of the graduates have multiple offers, as you can imagine, as product developers, engineers, hardware engineering, so on. We are in the biotech hub here in Gaithersburg, and so a lot of our students actually find themselves working in biotech companies in all sorts of jobs. So we've got very, very lucky in the last 3 years, the 2 years that we graduated students. We have graduated 2 magna cum laudes, and recently we have 1 summa. So this is the quality of the students we have, which is why we're so delighted to be able to offer this. There's another notable contribution. I don't want to boast too much here, but you see we have the fires in Canada. But we have a system built by our former graduate who is now in California, and that one is detecting wildfires in California, and they're working into developing things that will predict whether a wildfire will happen or not. And that's that's all within the purview of cyber physical systems engineering. So I guess with that, I think this is the end of my slide. If you want some information, very easy to you can Google basically cyber physical systems at Maryland, and we'll be the first to come. But if you wanted to directly go to the site, it's shadygrove.ec.umd.edu, and that is my my email. If you wanted to send a comment or so, feel free to do it. I'll be very happy to respond to you. Okay, so these are more, of course. That's it. Okay, well, thank you very much for your time, and I welcome some questions. Thank you.
You can entertain 2 questions, 1 or 2 questions. And yes.
So in terms of how many started and how many
could you please repeat the question? Sorry.
How many students are there in your program, and how many have graduated already?
Oh, we started small. So we graduated a total of 10 students in the last year or so, the last 2 years. So 10 students now, and we're in the past the pilot stage, and we're now ready to expand it to probably about 25 to 30 students in the next year or so. We cannot grow too fast because it's a very, very difficult course to scale up. But that is the current that's the current number of students that we have. How did we start? Well, this is not cheap. The first thing was to convince the State Assembly of Maryland of the need for this, and so we requested resources outside the tuition revenue, because the tuition obviously is not going to be enough to cover everything we wanted to do, and so we requested for what you would call startup funds, right? So this is initiated by the state of Maryland, and thankfully the the state assembly the delegates were very supportive, and so they just basically said, go ahead, and here's the resources to do it. And that is why they were able to not only build this building, but also provide the resources for us to get this off the ground.
Thank you very much. Is there an age limit for your applicants?
No, there is no age limit. As a matter of fact, I should tell you that one of our graduates he already had a degree from Brazil, and his degree is in pharmacy. So he's a licensed pharmacist in Brazil, and he came here, and he was just not very happy with the way that he is living his life. So he decided, I'm going to invest 2 years of my life, and that's exactly what he did. And he happens to be the designer of this thing I was telling you about in California, predicting wildfires. So yeah, that's a remarkable change. Now, he is not young. I don't know whether he would like me to divulge his age, but he is certainly about 45 years old. So he's old. He's older. But we do have very young students too. We have the freshman, which is only 18 years old. This is a new innovation from Maryland, where you get a degree in high school. You also have an associate's degree. So, and that's what you get. Anyway, so it's a large range of ages.
This one I'd like to.
Doc Mel, hello.
Yeah. Okay. Okay, thank you.
Hold on, one more question.
Okay.
Okay. Dr. Ramel, good afternoon. My name is Albert. I'm from Batangas State University. I'm an electronic and our president is also here. And if I'm going to pitch, for example, that course to be offered in the university, what would be a pitch that would convince, let's say, the president of the university to what's the delineation of cybersecurity with that of electronics engineering? Because I see some overlaps also.
Yes. Look, I imagine you're referring to Tirso Ronquillo, yeah?
It's here. It's here.
Tirso, are you there? Tirso, I've been meaning to contact you. Asan ka ba? Anyway, I remember, Tirso, how are you? It's a great pleasure to once again see you, no? I remember discussing something like this some years ago, no, when we had Musta, Tirso? Di ko tumatanda, but I think there's really a lot of I mean, we could probably do something very similar sa Batangas. One of the things that I love about your school when I visited it was the fact that you are probably one of the really very good mechanical engineering schools that at that time, I think you mentioned everybody passes the board exams. Is that still true? This is really remarkable. And plus, I saw Batangas State University as the leading national state school. So as far as this is concerned, the main issue here is, how do you leverage things that are you could get information online. We could come up with some sort of a relationship in which we could show you how certain things are done. But the important thing, I think, is to invest on the infrastructure. Thankfully, it's not very expensive anymore. Embedded systems are not very expensive anymore. And plus, you have a very nice, very clear mandate. Agriculture. I remember that we talked a great deal about technology in agriculture, and that is something that you could probably take a very important lead on using technology in agriculture. And so the long answer to the short question is it is doable, and let's come up with some ways to collaborate. We'd be very happy to share with you resources and some of the knowledge that we have so far in order to maybe promote a program similar to this at BATSU.
Thank you, Dr. Romel.
So ano ba, kakanta na ba ako? Pwede na bang kumanta?
We have 3 more speakers. So later I'll call you for the karaoke.
Sige, I'll be here. Thank you, thank you, thank you.
1 second. Yeah, can
Oh,
hello, Dr. Romel, can you hear me?
Yes, yes.
Okay, so we'll be extending our chat after this, no? And we'll extend our karaoke after this.
Sige, yes, this is Tirso, right?
Yeah.
Sige. I've been really wanting to have a good chat with you. Yeah, sige. Very good.
Okay, thank you. Keep safe.
okay, that was very interesting. Thanks. We now move on to the next speaker, Dr. Ralph Torinan, who will be giving a talk on integrative STEM to revive and sustain coastal marine resources and livelihood in the Philippines. Let me just briefly introduce you. Please allow me to introduce you. So Dr. Ralph Torinan earned his BS degree from UP and a master's degree from University of Rhode Island and a doctoral degree from the University of Puerto Rico. He had postdoctoral training at Florida State University, and he has been on Florida Tech faculty since 1995. His research interest is on the ecology and evolution of organismal. Beside invertebrates, uses the fish feeding system as a model with the goal of elucidating the roles of ecological and evolutionary processes in explaining organismal diversity. So I could keep on going on, but let us all welcome Dr. Ralph Torinan.
Thank you.
I can press the arrow button.
Which one?
Yeah, for this. This one.
Is that full screen?
Yeah, it's full screen.
Oh, thank you. All right, I'm very nervous because this is my first BAS talk. And so I'm nervous because I want to do it again. And so I want to qualify for my next talk, which is next year. So anyway, and the other reason why I'm so nervous is that it is very difficult to succeed so many great speakers. President of state universities and just distinguished scientists. And so the picture you saw a while ago was taken when I was 50 years old. And so I turned 60. I have my Filipino senior citizen card when I went home in May. And it was a wake-up call because when my wife was driving me to the airport to go to the Philippines, we started brainstorming as to what we can do. Because about over 40 years ago, there was a scholarship that brought us all to the University of the Philippines in Diliman. It was called the Ferdinand E. Marcos Scholarship Program for Fisheries. So that was not our intent for our career, including me. I was ready to go to the University of the Philippines in Los Baños with my meager NSDB stipend to take DS chemistry. The Ferdinand E. Marcos scholarship was twice that amount. It was about 700 pesos. NSDB then was maybe 250, 400. So we all shifted, and I am in the first batch of that. And so we started in 1980, graduated in 1984, but there was a vision of the old Marcos that never left my mind. The intent of the scholarship is to advance fisheries and aquaculture in the Philippines. And so 40 years later, it's more than about time to do it. So I'm going to share with you, I have a few, how I built my career, 35 years of research to finally go back home, and I did. Here's the reason. So I'm building up the rationale here from the global all the way to the local level. So we all know that our craving for seafood has substantially increased globally. The supply of seafood from the natural environment has substantially decreased over time. So there you go, is the deficit. Now there is a method of producing that has been largely underutilized, and that is aquaculture in the Philippines. But globally, if you look at that pink, it is our only hope to produce enough seafood for the 8 billion earthly humans that are now existent in the world today. But here is the problem. The second rationale is that the supply of seafood in the natural environment has diminished over time because of overfishing. So if you look at the income of fishermen and fishing villages in the Philippines today, they actually are going down and down and down. If you look at municipal and commercial fisheries, down by almost 2% in just a year. And it's never going to go up. But look at the potential in here, look at aquaculture. It is increasingly growing. But the problem with aquaculture is how do we deal with wastes from aquaculture systems? Because they have to be contained or managed so that we no longer bring more pollutants into the water in this case. And so, what I did was, as a part of my 60th birthday present, I went to the Philippines and went back to this area where I was 40 years ago before I went to college. And I was so humbled because the condition of the village actually is worse today than it was 40 years ago. So what are we going to do? We are trained scientists and researchers in the United States. So thank you, Dr. Villalobos, for inspiring us to go back and bring STEM to the Philippines, where it actually is much needed than anywhere else in the US in that case. So 3 years ago, there was this call for proposal by NSF to converge STEM into a way in which we can put all the minds together and come up with a product. So I called my friends from MIT, from the University of New Hampshire, from the University of Puerto Rico, Florida Atlantic University, and brainstormed. And we came up with this idea, which we called this NSF Convergent Accelerator. It was one of the programs to integrate, not just to work individually, but to come up with a package where STEM can actually produce one product in this case. And so we did, and we put together our own ideas and how, we came up with a way in which we can integrate all of this. So we have science. I'm a biologist, and so I take care of that. We have engineers, and technology and engineering are actually very related. A friend of mine from MIT, and they are going to make the design of this structure that we're going to make to produce fish, and we have our mathematicians to model where all of these wastes are going to go to predictably, so that we can take care of managing the waste in this case. And so there are computer models. So this product is what I now call SIMPAS. And you know what the trademark of a Filipino is? Acronyms. So, and I will explain what SIMPAS is, but it is a community-based ecosystem design, ecosystem engineered aquaculture system that could easily be transferred to communities in the Philippines. Where is its root? It is rooted in what we call in the US, Europe, Japan, and elsewhere in the developed world called IMTA, integrated multi-trophic aquaculture that is actually rooted in a very basic ecological principle, the food chain. So you have a producer of waste, nitrogenous waste, which is fish or anything that we culture, where do their poop go, right? It's wasted if we don't know, and if we don't integrate something in there that eats the poop, and in the natural ecosystem, there is. Those are our detritus feeders, our mollusks. And then, of course, what do we do with the nitrogenous waste? Well, nitrogen is one of the nutrients that make plants and algae healthy. So we integrate algae and plants into the culture system in this case. So there actually is a really nice flow of effluents, waste, to the first stage fixers, and then the final stage fixers before it goes back. There are two versions of this. There is also a natural field version of it, and that is what SIMTAS is actually based on, but both of them are rooted, take note, of the same principle, which is the food web principle in ecology. So the first system is what we call land-based. This is recirculating. And what you can see in here, this is our design, which is now being used in Puerto Rico. We have fish that produce waste. It goes into invertebrates like holothurians, sea urchins, and mollusks, any type of mollusk in this case. And then it flows into our photosynthesizing components, which are green algae, mangroves, seagrass, and so on and so forth. And then it goes back into fish. So nothing is left to waste in this case. The second system, which is developed at the University of New Hampshire, a friend of mine has done this, is the field version, which is our payao, our cages, right? And so it is the same system. You have all of the fish in the cage producing fish waste, and then it goes into the benthos, which will be eaten by invertebrates, and then it goes into what we call our flower garden, which are made up of green algae and all kinds of algae, as a matter of fact, and plants in that case. So what is SIMTAS? It is my innovative way of combining these two into a system. So it's called the coupled integrated multi-trophic aquaculture system. You can't see this, but for the first time, we're going to put it together in the Philippines. And what happens here is, for the early stage of the life history of a fish, they eat high protein diet, okay? And then what goes on at the grow out stage, the last 3 months, is what we call the adult food, which is now much more different from the larval stage. Now, if you put that in an ecosystem, there's a double whammy. There's going to be, that ecosystem is going to be disrupted at the early stage, and then 2 months later, it is going to be disrupted again with the introduction of the old fish food. So what we do is we grow them at the early stage in our land-based, and then transfer them to the fish cages only during the time at which they grow from subadults to the market size, a much shorter period of time. And the second advantage of that is, yes, there is going to be some input, but it becomes stable over time. So a new ecosystem is actually developed. Now, in the Philippines, especially because of our two-season system, right? During the non-typhoon season, we do both, right? And this is where our friends in engineering design is going to come in, is how are we going to make this portable, collapsible, so that during the typhoon season, we now tow our field cages to a much more protected cove and only produce in our land-based system in this case. And then go back out after the typhoon season is done, we do both in this case. And so that it becomes sustainable, our fishermen do not have to remake their pile after a typhoon in this case, because they can always bring them to safe, just like what we do with our boats, our yachts in that case. So we have our pilot study, which is based in the University of Puerto Rico, because I think it was, yes, because the Philippine government does not fund pilot studies. So I wrote 2 grants to NOAA. Thankfully enough, NOAA funded it, and so we have 2 ongoing. And this is to revive the fishing villages that have now been dwindling over time because of the diminishing catch from the sea in that case. So why Puerto Rico? It is my cynical reason, okay? Look at where Puerto Rico is, and look at where the Philippines is. You just go from the Atlantic Ocean and go, it is exactly the same environmental condition. In fact, because the Philippines is among the cradle of civilization in the marine world, all of the marine organisms that we find in the Philippines are represented in Puerto Rico. And this is where my wife and I live and where we work. It's only a 2-hour flight from Orlando to there. We can go there on the weekend, monitor our program. And now we are at this stage where we already, with my colleagues, we already have fine-tuned. So now I'm bringing it to the Philippines in this case. So where we are is to address this beautiful goal. I've lived with my wife now for 36 years. We've been together for 40 years. We have 2 daughters. And so I just love women. And you know what? Because in the fishing industry, women are in the backstage. They do all the washing, but they are better than that. So our curriculum is designed to teach our moms on how to raise food using our syntax, because we're now designing the informal education for STEM. With my colleagues in the education department, we are making them much more vernacular so that they won't suffer as I did for the earlier sessions, where it's all biochemistry and it's all about immunology and all that. What a way to really integrate STEM in this case. But here is the most beautiful reason why we do hope that it's going to work. It also addresses global issues, one of which is our craving for something that is resilient, and for something that is resilient is sustainable, and perhaps we can provide, maybe not at the availability that we have enjoyed throughout our lives, some to our future generations in this case. And of course, there is IMTA in there. And when I was asking myself about, where can we get the funding for this, right? So I went back to the Philippines, to my home province, Cagayan Valley, right? And I went to BFAR. BFAR has so many underutilized culture systems. They're there. Probably spent millions building them. So what I'm just going to do is convert them into an IMTA. And so they have all of these tanks. We're just going to be reconnecting them and refurbishing them in a way that the IMTA actually works in that case. So where are we? Fortunately, there is a new program that is just about to open, funded by the World Bank. Okay? And here we are, just waiting for that moment when they advertise proposals for funding. The Philippine government just received $209 million, but look at this, to specifically improve Philippine fisheries and coastal resiliency. And guess what is in that press release? IMTA. So we are ready with our package, because what I've been doing is helping BFAR come up with proposals. Unfortunately, we missed we missed Neda and IC by 2 weeks. I received the email when I came back here, and it was already mid-June, and we put together, we're ready with the technical aspects. It was just the communication between the regional and the central office, because it is mandated in that it has to be the central office that gives the blessing in that case. But anyway, we are ready for the next one in that case. So anyway, my dear friends at PAS, thank you very much for waking us up. And I want to mention two special women in our group this afternoon. They were the third batch of that scholarship. The two first ladies. The first lady of my household and the first lady of PAS. They were funded by the Ferdinand E. Marcos Scholarship Program for Fisheries. So there you go. We have the physical evidence that with our partnership with PAS, PASIC. I think I failed my first talk. Is it PASIC? PASIC, yeah. No, it's PASIC. So, anyway, thank you.
Inspiring, actually. So we'll first think about it. Because now you can pronounce. Yes.
Great idea.
Thank you.
So 20 years ago, there was a lot of talk about genetically engineered fish for improved feed conversion, growth efficiency. Has that taken off? And if so, is that applicable to what you want?
No, because genetically modified organisms really are not resilient for these communities. They will run out of supply. So there actually is transcriptomics that we integrated in this one, how to build resiliency by waking up all of those heat shock proteins that they already have. And so in fact, we had a publication last year identifying, and it shows that for fish that actually are more hardy, we call it in aquaculture, they actually have a much higher expression of HHP 70 and 90. But the thing is, they have to be awakened. So how do we awaken that? In our land base, while they are still young, we are going to expose them in variable sublethal stressors, which is very easy. Temperature. So and it's the easiest and the cheapest way to vary. So all we need to do is, instead of put them in an aseptic environment, we're going to train them for a month to respond to these changes on a daily basis by just changing temperature. And all we need is to wake up that HHP. Any environmental stressor after that, we are ready. So instead of GMO, we have natural, resilient building mechanisms that are already in the genome of this fish. So we were able to identify this, and I bet you that if we can include that as one of our criteria for selecting species for this system, it may work. Yes?
Yes, so I'm an applied social psychologist.
Thank you.
That principle that we always apply in starting community-based development programs is that we have to have a developmental component.
Yes.
You mentioned the women, right?
Yes.
And you're talking about an integrated STEM approach in dealing with all of these problems. My question is, where are the younger folks in this? What's the role of the students?
They actually are more fortunate, because I was talking to President Ronquillo, okay? One of the things that we started in Florida is to build built-in high school hands-on laboratory exercises. So they're okay, because they will receive that. It is our women in the backstage that need to be brought up. So we are developing high school laboratories. And in fact, I was telling President Ronquillo that the next time I go home, I can go to their nearshore lab. This is a very nice hands-on lab for our majors in marine biology, marine science, oceanography, and ocean engineering. For them to be able to design a collapsible payao instead of a fixed payao, they can move that and actually build an artificial reef that is actually close to being natural instead of just a bunch of stones that we now put out there. Look at this. Isn't that an attraction for ecotourism? You can teach these tourists that, look, if we do not disrupt this system by fishing and fishing and fishing, we actually build. So this would be a more attractive artificial reef than just a boulder of tires or cement and things like that, because it's complete. We have the predators, we have all of the detritus feeders, and then of course we have the photosynthesizing members that actually extract natural use waste and convert them into useful nitrate in this case. Yes, ma'am.
Sorry. My question is, all of this is great, thank you. He's my husband.
I invited her so that I don't have to explain to her why I have to go to the Philippines.
I have to put him on the spot. I'm a secondary school teacher, so you, me, Lenny, everyone else went to fisheries because of enticement of money, because we are all in need of that funding for education.
Right.
How, I mean, to be honest, we probably, and you just admitted that you were in a different field altogether. So we went there not because that is our passion, but because it was financially.
Right.
I mean, really, that's how we could have.
So, ma'am, what's the question? We're running out of time.
The question is, how will you entice Filipino children in the secondary school, even the university level, to go into this field, to be aquaculturists when.
Sorry to interrupt, but I think we need the expertise of Dr. Sandalamini. Yes, because this is, if we can package this into a revealing reality, kids are not that as we think about. We just have to inform them that if you care for your future, there is biotech in there. There is molecular biology in there. Can you imagine how we can compare the transcriptome of these that are being cared for versus those that are not being cared for, that are subjected to the bombardment of the disturbance all the time? It's beautiful to be able to explain that, look, these are exactly the same population, but look at their transcriptomes. These are the DNA that are actually expressed at a given time. That is interesting. Yes, ma'am.
So you were talking about this biosystem, and there are different species.
Components, yeah.
That's right. So will you change the balance of the aquaculture because they respond to different heat and changes in temperature, so you might change the whole. The balance of the system.
We intend to do that just at the conditioning stage, hormesis in medical sciences. So at that very early stage, that is where they're actually kept in that recirculating system. That's where we condition them. Once they go to the cage, they will remain at that condition for the rest until they go to the market and eaten by us. So yes, it's only during that early stage. But as soon as their their transcriptome is translated into protein, it becomes embedded into their system. And from our laboratory work, it can never be reversed. Which is one good thing about these heat shock proteins. Once turned on, it can be, it is there until the organism dies. So I think I hear you, but and thank you. I'll put that in my footnotes to make sure that we will we will consider that in furthering the idea. Yes, sir.
So how is this program sustainable in an economic sense?
Mm-hmm.
Is it reduce more, I guess? So that was my question.
So the existing system now is they produce one. They either produce fish or shrimp or clams. In here they produce three, at least three. And then of course the fourth one is seaweeds. Carrageenan is a global, it's in global demand because of all of the gel-based products that we need. So they actually quadruple their income. And with Synpass, which is a year-round, fishermen have a seasonal ability to produce. They only go out during the calm season. So during that season where it is Abagat or Magabat, or typhoon season when it's rough out there, they can take care of the land-based. So in year-round, actually, they have income. Yes, ma'am.
So the potential for the project as a pilot project.
Yes.
Is that ongoing?
Yes.
So what's the scoop?
The scoop is it is working, but there's always the ideal and the real. So what we are actually learning are the realities. So we have all of this conceptual framework and all that, but when we actually do them in captivity, the complication here is there are 4 organisms you need to worry about, and they have to be all alive. So we have an invertebrate zoologist with us. I am the fish biologist. I have an environmental biologist to take care of all the water monitoring. I have a botanist. It's really a cool STEM. I couldn't think of any other production facility that STEM is at work all the time. So we always communicated. We always communicated. Sorry. So yes, actually, that's where I'm studying a lot. I'm learning a lot from the Puerto Rico before I bring to the Philippines, because I'm discovering as many bugs as I could, and fix those, so that when we finally go to the Philippines, it will only be the local condition.
What about the collapsible?
That's why I need our engineers. It's a great idea, but I'm not an engineer, so I don't know how to design it. And that's where our engineering and technology minds actually can contribute. So they will solve that problem. Yes, sir.
Okay, so I would like to suggest something.
Sure, thank you, Dr. Achille. Yeah.
Maybe how to adapt the kids or the young ones to study about this, or fascinated about this. You mentioned about BFAR.
Yes.
And that there are some facilities that maybe BFAR and then talk to each other.
Yes.
BFAR set up a model like this and tell the vet, include that in the look of their field trips or whatever. So that the students will see and fascinated about this thing.
And that's where we the academe can facilitate. Government, BFAR with Chad. That's where we're going to need somebody like
something like Sentosa like aquarium.
Yes, absolutely. I think I overstayed.
After the session. So now we move on to the next speaker.
We'll bring you to Puerto Rico.
The last No, it's just first. Okay, Juice, I still have to introduce you.
Yeah.
Although everybody knows.
I'll make my presentation as fast as possible.
So yes, where is he now? We'll think about it. He actually volunteered to be one of the organizers studying next year's APAMS.
Wow. Yay.
Okay, so we now move on to the next speaker, Dr. Juice Santos, His talk will be on integrating household survey with interoperability input-output model of critical infrastructure systems, a case study of Hurricane Sandy. Let me just introduce him. Okay, so he is currently an associate professor in the Department of Engineering Management and Systems Engineering at the George Washington University. And his research interests lie at the intersection of systems engineering, disaster risk analysis, and economics. So he is a principal investigator in the 2018 Mitigation Saves report that has been cited by many journals, news media, and research agencies across the world. He is also one of the principal investigators in an ongoing multi-university National Science Foundation grant, collaborative research organizing decentralized resilience in critical interdependent infrastructure systems and processes. Without further ado, I would like to welcome Dr. Justine Tigno.
And then just use the cursor.
Yeah, either this one or you can.
Okay. Good afternoon, everyone. Here's the title of my presentation, and I just learned today that Peb, that's her name. Maybe she's already asleep in the Philippines now. I took the liberty of stealing the APAMS logo, which I thought was very, very excellently designed. So the cursor key there. So inspired by Mario's Born Band Nexus plot twist. This was a last minute change last night, so I was talking to Dr. Simmen so that I needed sleep last night. I had to turn off the alarm, and I needed 30 minutes more because I had to make a last minute change. So I was thinking the crowd who might be interested with Hurricane Sandy, I'm already writing papers on Hurricane Harvey and Irma with collaborators also from Florida and also Northwestern. So instead of that, I recently completed a paper. Recently completed a paper with DLSU on the 2020 Taal volcano eruption. So we spoke about fish diseases, and this is about disasters. So hopefully my presentation is not disaster. And to add to the element of the Born Band Nexus, how many minutes do I have? So I'm going to add an element of Mission Impossible here. So to give a chance to have more airtime to the last speaker. So we were very welcoming here in the United States. How many? Okay, I have my timer. So I'm so proud to have been also part of a big university consortium. We published the 6-to-1 ratio, which means that for every $1 investment in disaster mitigation, there's a benefit of $6. Inspired by Benjamin Franklin's, I know that everyone has heard of it, an ounce of prevention. I should say it right. An ounce of prevention is worth a pound of cure. Usually I mix it up. So I'd like to acknowledge Fulbright. The reason why I know something about autoimmune diseases, because when I was writing this paper during my 3-week stay at Makati Med last year. It's unfortunate, but it's also productive. So I was a Fulbright scholar last year, and I'm becoming nostalgic because I was a Balik scientist 2011. I returned 10 years later, and I also served as a visiting professor at the University of the Philippines. I did my Fulbright at LaSalle, and my professor at the University of the Philippines was actually mad at me, Aura Matias, because I did more public outreach to the LaSalle than my own alma mater. So anyway, here's just the agenda. everyone's familiar with the volcano. It's so beautiful, but it's very disastrous. So what I call inoperability here, it's the opposite of reliability. It's a simple number between 0 and 1. Reliability of 1 means the system is flawlessly operating. The opposite of that, inoperability of 1, you don't want that to happen, like skies falling, end of the world thing. So to cut to the chase a bit, the bottom line here is our model estimated 2.8 billion, anywhere from 2.8 billion to 3.5 billion. Now the estimate is higher. It's because we didn't include the inoperability, the direct losses suffered by the agriculture sector, which Calabarzon, Cavite, Laguna, President Ronquillo is also from the Calabarzon region. And this is something that data from Batangas was also what we used in this study. So stock and flow model, so disasters can damage buildings, physical infrastructure. But I think not everyone knows, like it's a lesson from the 9/11 case, is the business interruption, which is actually can be equally disastrous than the physical damage. So that's just the stock and flow. And what we use here is an economic model called input-output model, which won Leontief Nobel Prize in 1973. I really love economics. I actually had to take an extra economics course just to have Solita Monson as my professor back in the Philippines. So I already mentioned Leontief, and the primary reference, I also met the authors of this, what we call Bible of Input-Output model. So it's very hard to see, but probably one of the most practical and one of the strongest economic tools is the input-output model. It tells you that the multipliers contributed by each column sector. I don't know of any country in the world that doesn't have manufacturing as the biggest multiplier. So that's why every first week of the month in United States, everyone's so eager to know how many jobs were generated by the manufacturing sector. So in the case for the Philippines, it's the same. This is the 2018 input-output table for the Philippines, which just reveals also, validates the hypothesis that manufacturing is the biggest contributor to the economy. So the concept here is that disruptions to, let's say, electric power can cascade to electric power, water, transportation, where as a matter of fact, it can also lead to higher order or the so-called cascading or ripple effects, which is what we call business interruption. So let's say, just hypothetically here, you have a sector directly affected here. One sector is just minding his own business. He or she's minding her business, but it would eventually be affected because of the higher order indirect effects. So for Taal Volcano, we use the NDRRMC. It's quite a mouthful acronym. Somebody said that Philippines is a very fun, and I agree with that. Yeah. Also in the United States, the Department of Defense, you have lots of hundreds. So I also have a funding from Department of Defense. So that's why I had to escape from earlier today to attend the Zoom meeting. So we also used input-output data from the Philippines and also GIS maps. So here's the Calabarzon area. I actually climbed the volcano before it erupted. It's really beautiful. Highly recommended. You will see horses during the hike, which I thought was kind of animal abuse. Yeah, for those who have I once tried those horses because they're like the smaller types of horses, and I just felt that taking them along that precipitous path is not so acceptable for me. Just my opinion. So anyway, we have infrastructure disruptions here. Initial inoperability, which is just the opposite of reliability, and also recovery period for 3 main infrastructure systems for which we found data from the NDRMC, and it's tabulated here. And we used the input-output model to calculate the losses. This is not yet the losses, but what we did was we regionalized the Philippine input-output table. It's tough because we have a lot of missing data, but we have very good connection with MEDA. One of my the students who work with me has a very good connection with the NEDA, and we were able to come up with location coefficients to regionalize the Philippine input-output table that's suitable for the Calabarzon area. So here's one of the input data. So we have the roads. We actually mapped the roads for the affected region, and we found out that 3%, you might think it's very small, but actually it's quite significant for the region, like as high as like almost 100% inoperability for the roads around Tagaytay. But since we averaged it for the whole Calabarzon, so it ended up only 3%, but the duration of recovery is about 15 days. So here's the one for electric power disruptions, for which the average recovery period was about 6 days with a standard deviation of 8%. We are going to use standard deviation for performing uncertainty and sensitivity analysis. And finally, water disruption, it's not that much, 0.9%. So what we learned is Philippines is actually very resilient. So even those impassable roads like motorcycles, for those who haven't been to the Philippines recently, you would find that it can be irritable. But actually it's become useful during the pandemic period, like the motorcycles, delivering stuff to your home, Lalamove, Lazada, Grab. Unfortunately, there's no more Uber in the Philippines, so you have to change your app from Uber to Grab. So here's the summary. I already kind of touched upon this, so I'll go directly to the results. So we found out that manufacturing suffered the most, even for the pessimistic, even for the baseline as well as pessimistic, it suffered like a 1.1 billion loss here up to 1.4 billion loss. And for the whole region, Calabarzon, we estimated 2.8 up to 3.5, only for the losses directly triggered by the three infrastructure systems like roads, water, and electric power. So NEDA estimated 4.8 billion, and it's already been vetted. And we just our reasoning here is that we didn't include the direct inoperability to agriculture, fishery, and forestry. Although we didn't include that, we see that the agriculture, fishery, and forestry, this is just indirect loss. So even without the direct loss, agriculture, fishery, and forestry suffered. It's the top, it's among the top 5 sectors here. So here's, but what we are claiming here is that NEDA only estimated like one single number for each sector. What we did here is we actually presented the losses as they accumulate over time. So it's a dynamic model. So these are the conclusions. The paper can serve as a template for future studies. I don't know if I mentioned it, President Biden in our earlier mitigation stage, it's called mitigation stage, the 6-to-1 ratio. I'm actually very happy that President Biden actually cited the report for disaster, for emphasizing the importance of investments in infrastructure. So parting thoughts. So the reason why I'm adding it at the last minute is that this conference is about reimagining training of future STEAM participants. So when I was doing this, I was actually working in the Philippines last year, around this time of the year, with Tapia, Frederick Tapia, who at that time actually we didn't know that he would win. He would win one of the OYS Outstanding Young Scientist. So while we were meeting, in the middle of a meeting, he suddenly literally just jumped up and down because he got the message that he wanted. I also worked with Dr. Lamberta and Ms. Kristen Solis. They're co-authors for this paper. And I don't want to bore you with the details, but these are the things that they did. Dr. Tapia in particular took care of the hard part of the model, which I don't know if even to this day, I don't know if I can pronounce it. It's a Dirichlet distribution for uncertainty analysis. Maybe Edsel knows this. So here's us meeting at Wheezy, the Green Belt. And this is also the same day that Dr. Tapia received the news that he got the OYS award. So here's our Zoom meeting. So most of you already know Raymond Tan, who in real life is one of my best friends. And also, small world, Kate Abiso happens to be a student of mine at the University of the Philippines. And just a parting thought, I concocted this. I actually presented it to the Department of Education in the Philippines. I came up with something catchy. It's called Optics for Training the Future STEM. Optics stands for Science, Technology, etc., Engineering. Should be objective, first purpose, transformative, interdisciplinary. Convergent is a word that I borrowed from Coelho, and synergistic. And I learned of a word cloud app that's free, so I tried it, which shows GW, DLSU Philippines. I actually also wrote a COVID paper, which is a subject for the Philippines, which is a subject of a different presentation, maybe in Los Baños next year.
Thank you.
Just quickly, the reason why I switched from Hurricane Sandy to Taal Volcano is it's just my humble opinion that Mayon Volcano is even better than Mount Fuji. Maybe I'm just being biased, and it's erupting right now. So hopefully the damage will not be very bad. Thank you.
So, any questions? So, since you mentioned
Biomedical.
are you planning to also study
Mayon Volcano. Hopefully not, because the moment I studied, there must be
a
disaster. So hopefully it won't come to that.
According to them, it's very mild.
Mild, fortunate.
So what about Mount Pinatubo?
Oh, Mount Pinatubo. Actually, thank you for asking that question. This is not scripted. The very first paper I published was actually for Mount Pinatubo at the University of Virginia. I almost forgot about that. I'm getting old. And actually, I remember using a lot of data from Punong Bayan. Is that correct?
Yes.
So thanks for asking that
Yeah,
question.
it was, I visited there one time and I went to the building. It's just amazing, but the destruction.
I still have to visit Pinatubo because they say that the caldera that was formed, it matches the beauty of the volcano, but it remains to be seen.
Are you gonna do, did you work on Hurricane Ian here in Florida?
Yeah, actually, I'm collaborating with a student who already got his PhD. Maybe some of you are not from Florida, but it's a good university, Florida International University. So I'm collaborating with them. Like the main PI for this project is actually from that university.
I took note of your description. Beautiful but disastrous. So the local tourism industry, I offer another way of describing it. It is disastrous but beautiful.
Positive. I take that. I appreciate that. And because you were so popular this morning, I was actually going to ask a question more about feedback. I really salute you, sir, because I'm the head of the ABAC for my department, and my department alone, I already had like my hairline already received for one department, and for you to be able to have your whole almost entire engineering accredited, you must have superhuman abilities. And I actually acknowledge.
Talking about it, really is hard work to be it. But I would like also to acknowledge Joe Cruz, our Baase member here, past president, who also helped us and the journey. Instead, it's a journey. You really have to change the behavior of the people inside just to get it done. Thank you.
Yeah, thank you.
I don't like this question, but I was in the Philippines when this is happening.
Just before the pandemic.
And then we were supposed to have a class reunion in Tagaytay in the March, after the next month in March.
Lockdown.
And no, this is before the lockdown. So I was actually surprised because when we went to Tagaytay after the eruption, everything is clean. And everything looks normal. So I'm kind of surprised by your results because it looks like, but again, Filipinos are so resilient, right? It's like nothing happened and we were in the resort and you can actually still see Taal, the smoke coming out of Taal. But everyone seems to be normal. The tourists are there. Everything has been swept away. And it's oh no, I didn't go. We didn't go there. But I know you have to because I know you have a friend of ours. I didn't want to.
Yeah,
Very interesting. We may run out of water, run out of electricity, but we never run out of hope.
Did you do a study on Typhoon Haiyan?
Yes, Yolanda.
Well, I thought that one. And that we lived
More than up to now they don't know the exact. They just say upwards of 10,000.
But I have to say that they have recovered. I don't recognize. I went home, we sat in November, I went home in March. Corpses were still all over the place. So we thought we would never recover. But as you said, we are resilient and the Taclobanans.
Just to react on what you said, just very, very quickly. I know there's a fourth speaker, and Leia. The day after a disaster, maybe a few days after a disaster, like the news coverage would see Filipino people laughing and smiling. As a matter of fact, we were working when we were still young graduate students of UV, University of Virginia. Me and my friends were laughing, and the Americans told us it's a constructive criticism that in every sentence that we say in Tagalog, at the end of the sentence we laugh.
So period.
That's one thing that I don't want to let go of.
Yes.
Whoever said hope, somebody said
President Hercules.
Oh, I'm from the Pinatubo area. So I was not there when it happened, but that was 1990, right?
Yes.
So that's 30 years, at least 30 years. And I think that the region actually is much better now than before Pinatubo. I mean, economically.
Yeah, I'm good.
Yeah. So big in.
Do you want to put your laptop here?
So big international airport.
Yeah, and Clark also. I mean, I think it actually turned out to be good that the Americans were chased away by the.
Yeah, I remember. I was still in UP and they were saying, like in Tagalog, patalsikin ang US Embassy. And then Pinatubo happened. I have very. I have good memories of Pinatubo. I was with my mother. I was in a dormitory in Kalayaan. We're buying an electric fan because it's so hot. And then at 3 p.m., it suddenly turned pitch dark. Unbelievable because of Pinatubo.
Thank you again, And last but not the least, I would like to welcome Engineer Albert Amance, who will give a talk entitled, I have to wait, and our tour guide, a cultural adaptive mapping platform using mixed reality. Can I just give a quick introduction? So Engineer Albertson Amante is an electronics engineer and the Vice President for Research Development and Extension Services at Batangas State University. So So the National Engineering University is serving since 2017. So with direction from the university, Engineer Amante also spearheads the development of the innovation programs of the university, which includes the establishment of the university-based technology business incubator, the Center for Technopreneurship and Innovation, the Innovation Consortium of South Luzon, and the country's very first Knowledge, Innovation, and Science Technology, KIS Park, a PESA facility which the university is developing. Let us all welcome Engineer Amante.
I think I'm the last speaker. So, first and foremost, I would like to, of course, thank two presidents who are here. First President Mariano San Domingo, yeah, for the invitation, and of course to our university president for signing my authority to travel. Thank you, press. Okay, so my talk is about a project that we completed actually during the pandemic. Like the other presenters, I also used an acronym here, but mine is very short. It's only MR. This is actually Mr. Tour Guide. So MR actually stands for mixed reality. No? So Mr. Tour Guide is a mapping platform that makes use of mixed reality. So this one we completed, of course, with generous funding from Bat State U and the Department of Science and Technology, PSHER, under the creative industry sectors. They're working on the creative industries as well. They're funding the creative industries. The creative industries is quite broad. Would you believe even yung mga sapin-sapin is part of the creative industries according to DTI? I don't know. Yeah. Okay, you can actually scan the code. That's the website that we initially developed for this one. So just to give an overview what MR is, what mixed reality is, how is it different from augmented reality and virtual reality. You have a taste of it yesterday actually with the talk of Professor Eric Sands, and when I heard of his talk, we have things in common that we are doing, and I was very excited also to visit his facility. Imagine having clients, electronic arts, EA. Millennials, are the millennials here? Okay. So yeah. So what's the difference between augmented reality and virtual reality? So augmented reality makes use of an immersive platform. It can be in a real world, in a virtual environment. You can interact with both worlds and clearly distinguish between them. You really just have to, let's say, make use of your cell phone or your mobile devices. If you're familiar with Pokemon Go, millennials, where are you? Okay, okay.
My daughter.
Yeah. So you get to play using mobile phone in a real environment. You open your camera and there will be Pokemon popping out. Those are augmented objects. So that's why it's called augmented reality. On the other hand, virtual reality actually makes use of an entirely virtual world. Yeah, for you to experience that, you have to make use of a headset, virtual headset, or VR glasses. No? But what's the difference? So what is MR? So MR actually is a combination of mixed reality and augmented reality. So this is what we did in the platform that we developed, which is Mr. Tour Guide. But what's the motivation? Of course, like what I said, we implemented this during the pandemic. So it became a 2-year project actually, and it's funded by the UST PSHER. The motivation is for the tourism industry. In 2019, that is pre-pandemic, the tourism industry in the Philippines really is booming. Imagine 8.26 million foreign visitors and $9.31 million tourist receipts. So that's 128 daily spending per average foreign stay. And everything, all of those values are gone when the pandemic came. In fact, based on the data from the Department of Tourism, it affected 50,000 jobs, which is equivalent to around 5.4 million labor force. So the idea of the team is to actually develop a platform that would encourage the people after COVID-19 would subside to really be interested in going back to the Philippines. No, make them be excited. Of course, support the tourism industry so that after COVID will subside, because we're positive during that time that it will be an endemic eventually. So people would come to the Philippines back again. But as we are developing the platform, we see another opportunity as well, because this may not only be a platform to promote the tourism industry, but it can also be a platform for digital archiving. Like what our good friend mentioned just a while ago, the Philippines is very rich in natural resources. We're also very rich in natural disasters. And one of which, for example, is earthquake. So last year, 2022, the northern part of the country was hit by a magnitude 7 earthquake, which badly hurt some cultural heritage sites. I'm talking about the Agripine Church, Beacon City, which is actually a UNESCO World Heritage Site. Imagine that. And a couple of old vintage houses in Vigan and Ilocos Sur. If you are visiting that area, some of them actually are now gone. No? So look at that. We don't have a footprint. We won't have a recall of that place if it becomes destroyed, if it is destroyed, right? Also, just this year, there was a fire at the Central Post Office in Manila. Now, the Central Post Office in Manila is actually a cultural heritage site as well. As a cultural heritage site, it means that it has some cultural value, no? That we should somehow preserve. But things like that can happen. So this is where also the platform would come in. It can serve as a platform for digital archiving. So imagine this fire, which lasted around 7 hours. And it happened during the night. And this is where the national IDs, I heard, were being printed during that time. So that's why perhaps if you apply for national ID, it's delayed because it probably have been toasted. Okay. So what is Mr. Tour Guide? So Mr. Tour Guide makes use of a locally developed mixed reality platform to provide safe and immersive experience of different cultural heritage sites and tourist destinations. We initially implemented this in the province of Batangas, and we're ready to roll out in the entire country to map out cultural heritage sites in all provinces, hopefully. Users will be able to visit the site at the comfort of their home, resulting to increased interest in heritage and culture. I'm also here to promote culture in Batangas. That's why I brought a couple of bees. Thank you for complimenting my barong. Ito po ay Taal, may from Taal, Batangas. If you're interested, I can help you. Probably, sabihin niyo lang po, small, medium, and large. Ang po dyan, kailangan ako po mag-deliver sa inyo. Of course, this one is pinya.
Wow, pinya.
If gusto nyo po ay sabihin natin medyo high-end ng konti, you can look at President's Barong. That's we call that full calato. That's full calato. Those types of tradition, those cultures, that type of barong takes 1 year for a single sewer to finish. 1 year.
Adi ang mahal, sir.
It is.
Very expensive, though.
I have a price list if you want.
Papa, did you bring something for the girls?
For the girls, usually we have this bolero that you can wear.
Yeah.
Sabi ko siya, dapat meron tayong mga best dress pagkabaan.
We'll do that. Next year, we'll do that. Okay? Yes, magbarang-barang tayo.
Okay, so look at this. So this is actually the methodology. Here we have a 360-degree photo,
picture taken at different locations per site using simple GoPro Max or 360-degree camera. Now, so many photos of the site are taken in different locations. So the images were used as data in building the 3D low poly replica of the site. So what we are happening here is we're creating a digital twin of the actual site, making it more on the virtual reality side. And I'll explain later where does augmented reality comes in. So using a 3D graphic software, the images are projected into a low poly 3D representation of the room. So kumbaga, if you have a canvas, kung nakakuha kami ng 360-degree images, what we do is we design let's say a 3D replica of the room, and we somehow paste these photos there, and then we do the stitching. That is where the image processing comes in, and the stitching. So what's the difference between what you see in Google Earth is that ours is much smoother. You won't see any, what you call that, distortion, something like that. Okay, so we're able to optimize that using the algorithm. So each room is then stitched seamlessly together using the location of each camera. So the 360-degree photos are then baked. The term is baking into the low poly 3D representation of the building. Baking the photos in the 3D representation of the building creates texture materials which will be interchanged depending on the position of the user. And we were able to integrate this in a website, because which is quite unusual, because having it in a web platform would require some optimization. Usually mixed reality platforms not available online, rather it's available offline because of the memory it consumes, but through proper programming of the team we were able to come up with an optimized system that can handle things. So like I said, we were able to also develop some interactive virtual maps. The icons that you see here are developed in-house by the team. Even the logo that looks like bitaw, pero hindi po sinasadya yun. Ano po? Okay, so these are actually we have I think 12 initial sites in the province of Batangas, most of which are churches. In fact, some are ngayon ko lang nakita na meron palang ganun sa Batangas. Okay. So some matrix manipulations. So since there are many textures in a single site, the images will be stored to an array data structure as shown as matrices. The matrix shows a general structure of the image array inside the program. The M corresponds to the number of objects. It can be walls, it can be tables, it can be chairs that were captured, and the N refers to the number of 360-degree images taken on the site. The bigger the site, the bigger the more images that we will be needing. So the matrix shall contain all the necessary image of each location inside a single tour. It handles the storage and calling of all image textures based on the user interaction. So the simple flowchart is shown here. I call it simple because what it does is that there are key points that you should, these are white circles that you can simply press, and then you'll be transferred to another location. So that's will be transported into that area, and you'll be able to see another space. So when the user clicks the hotspot or that white circle, a function will run the algorithm and selects the correct image texture based on the clicked hotspot information. So it's as if you are navigating. You can use also the desktop, but it's better experience if you're wearing a headset. Unfortunately, I was not able to bring one. But let me just share with you, let's say, this is the desktop view of a church. This is an actual church in Laurel, Batangas. It's called Simbahang Bato. So notice that there are actually white spots there. It's how you navigate the system. No, it's like the 360-degree image of the sites. We were also able to embed there a couple of some augmented reality features, like some important artifact that can be highlighted as well. If you go to the website that I've presented using the QR code later, you'll be hearing also some audio effects. So that one is some augmented reality feature, making it more interactive. A good way also to, let's say, to teach cultural heritage sites among students, right? Okay. So another demo I have is also we're also able to map out the Mabini Shrine. It's a museum dedicated for Mabini. This one is located in Tanawan, Batangas. So we're able to showcase the museum dedicated for Mabini. Notice that, yeah, like what I said, you use that circle as a way to navigate around, and those are augmented reality features, so you can highlight those artifacts. Okay. So in terms of technology roadmap, we intend to still make use of continue developing this using other technologies such as digital twin. Just yesterday, I received the news that our proposal to develop parallax is approved by the UST TAPI. Now, this is learning from what we had or what we developed in Mr. Tour Guide. We're going to come up with the development of some virtual laboratories for engineering as well as for STEM, because that is, I think, a gap, because investing on laboratories can be very, very expensive. Let's say if you have a chemical engineering program and if you would like to experience to your students an environment, let's say, of a chemical engineering or a chemical plant. So through this platform, just wear your virtual reality glasses and be able to somehow be as if you are in the actual environment. And we're planning also to develop some exercises so that the students or the learners would be able to solve some real life problems. We're looking at also at case studies with the industry. This can be used by the industry, let's say, for them to train fresh graduates, for them to at least to have a clear understanding of how a real environment, let's say, for a semiconductor industry would look like. So, so that's the plan, and of course for STEM as well. We also intend to make use of digital twins to have a more, should I say, realistic representation or archiving of these cultural artifacts for preservation. But we'll start with small objects. So this is Parallax, the one I was talking about. This is a mixed reality platform for teaching STEM. We're set to develop modules for the engineering, for the STEM, and for an industry partner. So these virtual engineering labs can be viewed through headsets for a more immersive experience. There will be some customization as to what particular field or what industry partner we're going to have. And it will serve as a training platform can be a training platform for new engineering graduates. Likewise, we are also making use of haptic feedback. These haptic feedbacks shall be in the form of gloves, so that you won't just simply see or look at let's say, an artifact, but you'll be able to feel, like how soft it is, how hard it is, how hot or cold the artifact is. Okay, so that is through haptics, and we've designed a couple of platform making and tethered and for remote applications, meaning it is wireless, and we still made use of WebXR as a prime interface for this application. Okay, so I think this is my last slide. I would like to acknowledge, of course, the team. We are composed of very young millennials who are, we actually collaborated, and we're from different backgrounds. I'm an engineer. We have someone from, of course, whose expertise is on computer science and programming and even design. And we have on board someone who is an expert on culture and arts. Okay, so that is Mr. Tour Guide. Again, you can actually visit the site. It's just scan the QR code. Thank you very much.
Yes, Mario.
Yes, I'm involved in physiology, local physiology, And in the past 2 or 3 years, we've been trying to form an aggregation of local historical societies across the province of Rizal. They would really be interested in this because right now some of them are doing cultural mapping.
Yeah, correct.
But this particular technology would probably fast track.
In Rizal, what's that, sir? What's the local museology in Rizal?
No, in Moro we have the Museo Mambayan of Moro.
Okay.
There's one in Antipolo. They're actually creating
Ano museum? Yeah.
in Bililla. So there are 13 municipalities in one city.
Okay.
But my questions, I have 2 questions. What is the cost, of like adapting or adopting this particular technology? And number 2, what is the learning curve, for someone to be able to get trained in using this technology?
Interestingly, the technology actually is not novel. The novelty comes in perhaps on our end, we're able to optimize it somehow using some algorithms that we used. But we actually had implemented also in parallel to Mr. Tour Guide is virtualtours.ph. That's under the British program that President Payne mentioned. What we did is we partnered with some universities because it's pandemic during that time. We partnered with some universities. We identified tourist sites. We simply gave them the instruction on how to get the data. And they were able to collaborate by them sending the data, and we'll do the processing, and we'll eventually do the databasing of these sites. And it works. So aside from Batangas, we have also a couple of sites initiated in the north and in Visayas. So in terms of data gathering, really collaboration helps because we are only in Batangas. And considering that, for example, state universities and colleges in the Philippines are strategically located, almost all provinces are represented, then we can actually tap in, share with them how to gather data. The university shall be the repository, and we'll do the final enhancement, and will ensure the sustainability of the program. Yeah. Okay.
How long did it take you to do? Is it all of Batangas already that you've been able to do the?
Yeah, so only now in Batangas. So we for a year we're able to scan Batangas, but that includes the R&D aspect of it. Yeah. So, but for now, I think I feel to answer the other question. How much is that? The component actually of the DST TAPI program is to determine its valuation, but somehow we're able to come up, let's say, of an estimate on how long we can accomplish, let's say, a specific site. It will depend, of course, on the area. But let's say for the one example there, the Simbahang Bato, it just took us around a week to develop the virtual site. We call it a walkthrough virtual environment. And we are also coming up with some business model, because I'm into entrepreneurship and innovation. We somehow have some business model. We've developed a business model in Canvas for this, and how we can perhaps, let's say, cater to possible clients, like, for example, local government units, or even private industries, or those, let's say, who are in the construction, no? Because in the real estate, no? Because probably they would like, let's say, to have for the clients to have an actual experience of a building or a house that is being built for them, something like that. So those are possible applications of this platform. Yeah.
I just heard, and I'll connect it to others. I just heard yesterday that the new slogan for DTI is Lab Philippines.
It's DOT. DOT.
Yeah, I need more coffee. Correction. I prefer the previous one, which is more Manila, Philippines. But having said that, do you think this would also be that the DOT would be a very
Because I'm looking at your website right now.
Correct.
It's really cool.
We initially partnered, sir, with the Batangas Provincial Tourism and Cultural Office of the Batangas Province. So that is actually a part of the agencies under the Department of Tourism. We've pitched this already to the Batangas Province as well as other municipalities in Batangas. We did some validation, and they are very much liking the platform, and we have now plans with them and integrating them in their own websites. So this would also be, of course, we wish to have this integrated at a national level, and the Department of Tourism really would play a vital role for that. We also have talks with the national NCCA, National Commission for Culture and the Arts, because they actually have the mandate on the preservation of the cultural heritage sites. Yeah, so for the Parallax program, because there's also a component of culture and the arts, they agreed to be a partner as a resource person for that particular project. Yeah.
And then just really quick question, answer all the yes or no. Are you the inspiration for that?
Parang wala pong balbas. May pangis daw po. Ako wala. Sabi ni Sarah.
Last question.
So I'm amazed. This is great, as well as just a little, maybe an idea. I was really also amazed at the presentation last night at gaming, because we are both amazed at our 5 and 3-year-olds who can do more than we've ever done. But wouldn't it be nice if we did, I know he was talking about Madden games. We have badminton, ping pong. I would love to see our local games be on digital platforms. But my question really is, how would this compare to YouTubers going to the sites, showing the sites, and still have the digital component? It's still archiving it, but live. So if you were to pitch this to, a government, how would you really sell this compared to a living tour guide?
Yeah, of course, nothing would replace a true and living tour guide. But as compared to, for example, a YouTube live, of course, the YouTuber will not be always there forever. But this one, you can actually have it preserved. And it has a different view. So you'd have a different perspective. Like what I said, of course, when you look at YouTube, you're just looking at to where the camera is pointing. But with this one, you have actually the control. You can look at the floor, and you look actually at the actual tiles. You look above and you actually look at the trees, for example, or the actual ceiling. And if there would be birds flying, you would be able to hear it. That's the dream. Or if there would be, let's say, a drizzle coming, you'll be able to hopefully feel something like that. So I think that's a valid proposition. Yeah. Thank you very much.
We have a wonderful session this afternoon. Let me just give the certificate of appreciation to the speakers.
I just love his smile.
Sorry, Mark. So you can ask questions if you have more questions while we're waiting for it. There's a yes, there it is. So, the certificate of appreciation is awarded to Rommel Gomez. Next slide, please. Ralph Torinan,
Juice Santos, Engineer Albert Amante, in grateful acknowledgement for imparting their valuable knowledge and insights as oral speakers in the 2023 annual PAASE meeting and symposium with the theme Reimagining the Training of the 21st Century STEAM Workforce, held from June 26 to July 1, 2023, signed by Dr. Mariano Santo Domingo, PAASE President and 43rd APAMS co-convener, and Dr. Justine Tigno as the co-convener. Again, thank you very much.
Some announcements. Tonight is a free night, so you can meet with your relatives, your friends, etc. There is no formal program tonight. Tomorrow. Yes. Okay.
And tomorrow we'll just check, some of the information we received, just to confirm about your plan to join or not join us on Saturday, because we would like to know whether you have arrived, and so we can accommodate you, and also whether you need to leave or depart, from Orlando International Airport on Saturday too, so that we can, make sure that you get to the airport in time for your flight. So thank you very much and enjoy your dinner. We have water here. Please take some to your room. You pray for that.
Para mag-official naman ba?
I will just